Thank you.
College is often viewed as a mere institution, a labyrinth of classrooms, textbooks, and endless exams, where our future is shaped. Yet, when we step foot on campus, we realize that we are not alone on this journey. University transcends academia, transforming into a vibrant community where individuals unite. To learn from one another, to grow and to forge unforgettable bonds. It's not always about having a precise plan. It's not always about figuring out. It's not always about reaching the finish line first. It's about discovering what we are truly passionate about. The true essence of college, however, lies beyond the confines of academic pursuits. What truly makes college so special is the life that's lived within its walls. It's about seeing things from different perspectives, exploring unique possibilities, taking on new challenges, finding joy in friendship, embracing individuality, expanding our knowledge and skills, and immersing ourselves in extraordinary experiences. It's a path that teaches us about resilience, courage, and determination, a quest that allows us to pursue our goals with purpose and clarity, a quest that helps us find balance, happiness, and fulfillment, a transformation that inspires us to live with intention, curiosity, and joy, a trail that leads us to a deeper understanding of who we are and what truly matters. It's a journey of self-discovery. Every experience shapes us into the best version of ourselves. It's a college for education. What we live is a treasure trove of experiences. Experience a whole new world of academic excellence in a global environment. Many places today.
Good afternoon, students. I welcome you to 29th day of Daya online CET NEET coaching. Now we can say CET coaching. Dear students, today first session you are going to have biology of Professor Priyanka. Next we are going to have chemistry of Dr. Ravi Shankar. Now you are going to have a session, the revision and mock exam paper discussion. I hand over the session to Priyanka madam. Please take over and share your screen, madam.
Yeah, good evening, sir. Thank you.
And madam, students will put their queries in Q&A box. Please address them.
Yeah, sure.
Thank you.
I hope it is visible. My voice is clear.
Yes, madam. Screen is visible and voice is also clear.
Good evening, students. So today we are going to see the revision of first PU. So have taken a few questions from the previous years. We are going to discuss with that. Okay, so then we will see further. So I have taken previously one more teacher has done few more years. So I'm taking from 2019, 18, and 17 questions of first PU we are going to discuss today, right? So in that first question, this question has seen in KCET 2019. The first question is, which of the following protozoan parasite causes sleeping sickness? So we are going directly with the question answers here. So you are going to tell about the answers. We are going to discuss directly question answers. So I hope the first answer, which of the following protozoan parasite causes sleeping sickness? The options are Plasmodium, Entamoeba, Entamoeba, Leishmania, and Trypanosome. So which is that which causes sleeping sickness? Right. I have got two answers. Anybody else? So you can read the question again. So I'll help you out. Which of the following protozoan parasites causes sleeping sickness? Is it Plasmodium, Entamoeba, Leishmania, Trypanosome? So yes, I have got answer as D here, right? So let's see whether the answer is right. Yes, the answer is right. Why D? A little bit of information regarding trypanosoma we will see here. That is, whatever flagellated protozoans are there, they are the members or this flagellated protozoans. They are either free living or parasitic in nature. And why they are called flagellated protozoan? Because they have flagella, as the name itself says. Among them, the parasitic form of protozoan causes sleeping sickness. The best example for that is trypanosoma. So I hope why trypanosoma and what is it related to? I hope you got it, right? So we will go with the next question. So if you want, I'll repeat it. So trypanosoma are the group of flagellated protozoans. Okay, in the flagellated protozoans, they will be either free living or parasitic, and parasitic one will cause sleeping sickness. So the parasitic protozoan is the trypanosoma, which causes sleeping sickness. I hope it is clear now. So we will go with the second question. Which of the following phyla possess body cavity as shown in the diagram? So here there is a diagram which is showing the body cavity. Which phyla belongs to this group, or which phyla shows this kind of body cavity? Is it Annelida, Porifera, Ascalminthus, or Coelenterata? So I'll repeat the question. Which of the following phyla possess the body cavity as shown in the picture? So that is either Annelida, Porifera, Ascalminthus, Coelenterata. You have to check the picture and let me know what is the right answer. Sneha, is it A or C? Which one? You have told both. So C, okay. C, Anvita is telling C. So let's see the answer then. So answer C is the right answer. That is Ascalminthus. Why Ascalminthus? Let's see here. So the body cavity in some animals, if you see, the body cavity is not lined by mesoderm. Instead of mesoderm, is a like there is a mesoderm present as a scattered pouches in between the ectoderm and endoderm. If you see the picture here, right? So that body cavity, we call it as pseudocoelom, or the animals which possess this is called as pseudocoelomate. So if you see, this is ectoderm and this is endoderm layer, and this is the mesoderm layer, where the whatever the layer is there, mesoderm layer, they are present as a pouches, scattered pouches, where and they create a body cavity here. So it is a false body cavity, hence we call it as pseudocoelom, and the animals are called as pseudocoelomae. And Ascalminthus are the the group or the phyla which has this kind of body cavity or which possess this kind of diagram. I hope the answer is clear. We'll go for the next. So the third one, the trees growing in temperature regions show clear demarcation between spring wood and autumn wood. This is because the climatic conditions are uniform throughout the year. The water stress is more, the temperature is high, the climatic conditions are not uniform throughout the year. What could be the answer? The trees growing in temperate region shows clear demarcation between spring wood and autumn wood. This is because why? What is the reason that in the wood you can see two colors, right? One will be a light and one will be a dark color. Manu is asking, ma'am, for CET exam, you should write biology exam. Yeah, that's why you're attending the classes, right? I hope that's the reason you're attending.
Dear students, madam, just one clarification I wanted to do. Dear students, sometimes what will happen is PCM, if you are not eligible, sometimes biology will also be considered, even engineering ranking. And BSc Agri ranking, PCMB will be considered, all for your board as well as your CET. And all other allied health science and nursing, anyone who is going, nursing as it is, separate exam is made, allied health science or any alternate this thing, you need to write biology, and attending biology is mandatory or good. Anyone who is very clear on engineering, on very confidence on PCM, you can choose. But better, my advice is write all the four. Biology should also be written. Thank you. Thank you, madam.
Yeah, thank you. I hope the answer is clear for Manu. So the question, we'll come back to the question. The trees growing in temperate regions show clear demarcation between the spring wood and the autumn wood. This is because what? What is the reason? We have got only one answer. Anvita has given D. The climatic conditions are not uniform throughout. Any other answers for this? Okay, Sushmita is also telling D. So let's see the answer. The answer says it is A. The climatic conditions are uniform throughout the year. Is it the right answer? Let's see the reason. So in plants of temperate region, cambium is more active in spring and less active in autumn seasons. So in the temperate regions, climatic conditions are not uniform throughout. However, in tropics, climatic conditions are uniform throughout the year. So here, so what we are talking about is the trees growing in temperate regions. Sorry, it has given temperature. Temperate regions show clear demarcation between spring wood and the autumn wood. This is because the climatic conditions are not uniform throughout. So sorry, this answer is wrong. Your answer should be D, right? So this will be the right answer. They are not uniform throughout. Hence, you can see the clear demarcations, right? So the next question, identify the part labeled as M in the given diagram. So you can see the diagram over here. We have labeled one part as M. You have to identify what is M. It is chromatid, kinetochore, centromere, or satellite. What is the right answer? You have to see the picture properly and identify the part which has been labeled as M here. So it can, what is it exactly? Chromatid, kinetochore, centromere, or satellite. So what could be the answer? Satellite, is it satellite? D. Muhammad Ashif says it is B. So let's see the answer here. So the answer is D, the satellite. Why? The reason, a few chromosomes having non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called a satellite, and here M represents that satellite. So what is happening? Usually what happens is, in few chromosomes, there will be small constriction at the end of each arm, or the constant, at the constant location, you can see a small secondary constriction. So this small fragment, we usually call it as satellite. So the answer here is the D1, right? So what is the function of protein GLUT4, G-L-U-T, GLUT4? What is the function of this protein GLUT4? It acts as an enzyme, enables glucose transport into the cell, fight infectious agents, functions as intracellular ground substance. So I hope you remember this. What's the function of protein GLUT4? So either it acts as an enzyme. What is its function basically? The question is, what is the function of GLUT4? Enables glucose transport into the cell. Is it the right answer? So Anvita, Shushmita, and all, Sneha says it enables glucose transport. So let's see. The answer is B, right? It enables the glucose transport into the cell. Let's see a little bit regarding it. So GLUT4 is an insulin responsive glucose transport. So GLUT4 is nothing but an insulin responsive glucose transport that is found in the adipose tissue and the brain. So what it helps in, it is present in the cytoplasm of the cells in vesicles, from which it is translocated to plasma membrane under the influence of insulin. That is, under the influence of insulin, these they get translocated from the vesicles to the plasma membrane and helps in delivering the glucose, or helps in transporting the glucose across. So that is the function of GLUT4. Sixth question, cell in the quiescent stage, G0 stage, what do we say? Always becomes cancerous, show indefinite proliferation, remain metabolically inactive, remain metabolically active. So what do cell do in the G0 stage, that is quiescent stage? So what could be the answer? So as I said, you please put the question number with the answer so that it won't be confusing. So cell in the quiescent stage always becomes cancerous, shows indefinite proliferation, remain metabolically inactive, or remain metabolically active. What is the right answer? So you all say it is D. Let's see what is the answer. Yes, D. What could be the reason? So let's see things about it. So in the G0 stage, what happens? Some cells in the adult animals do not appear to exhibit division. Means they don't show any division. Example is the heart cells. And many other cells divide only occasionally. So these cells that do not divide further exit G1 phase to enter an inactive stage called as quiescent stage, or it is also called as G0 of the cell cycle. So cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending upon the requirement of the organism. So what you are telling, I hope you understood. So G0, in G0 stage, or in the G0 condition, so some cells, they do not divide to further exist in the G1 phase. So they will remain inactive, and that stage is called as quiescent stage, or the G0 stage of the cell cycle. So in this, the cells remain metabolically active, but they will not proliferate. They will not divide unless and until the requirement is given by the organism's body. I hope the answer is clear. The next question, consider the following statement, that is 1, 2, 3, whatever is given below, regarding criteria for the essentiality of nutrients in plants. So the first statement, the presence of elements is must for plants to complete their life cycle. The role of elements can be replaced by another in their life cycle. The elements must be directly involved in the metabolism of plant. So which statement is true regarding the essentiality of nutrients by the plants? So which statement you will say it is true regarding the essentiality of the nutrients in plant? Is it presence of elements is must to complete their life cycle? The role of elements can be replaced by another, or the elements must be directly involved in the metabolism? Which statements are right? We have got two answers. A. Any other answer? Any others to be answered? All right, so let's see the answer. Yes, the answer is A. So let's see what is it. So the criteria essential of an element are given. Essentiality of an element is given. The element must be absolutely necessary for supporting normal growth and reproduction. In absence of that element, plants do not complete their life cycle or set the seeds. And the requirement of that element must be specific and cannot be replaceable, not be replaced by any other element. In other words, deficiency of one element cannot be met by the another element. An element must be directly involved in the metabolism of the plant. So these are the three criteria for the essentiality of an element for the plants. And according to these three, only 1 and 3 will be the right statement given here. The second statement is the wrong one. So option A is the right option. 8th question. During the chemoosmotic synthesis of ATP in photosynthesis, the protons accumulate in the intermembrane space of chloroplast. The proton gradient is not required. Protons accumulate in the intermembrane space of the mitochondrion. Protons accumulate within the lumen of thylakoids. So during chemoosmotic synthesis of ATP in the photosynthesis process, what is happening? What could happen? Is it protons accumulate in the intermembrane space of chloroplast? Proton gradient is not at all required. Proton accumulate in the intermembrane space of mitochondrion, or they will accumulate within the lumen of thylakoids. Where do the proton accumulate? If not, they are not required, or what? What is the right answer? So during chemoosmotic synthesis of ATP, that is adenosine triphosphate, what is synthesized in the photosynthetic process, where do the protons accumulate? So the name itself says photosynthesis, chemoosmotic synthesis. And the answer is C. The protons accumulate in the intermembrane space of mitochondria. Let's see why. So during the chemoosmotic synthesis, ATP is linked to the development of proton gradient across the membrane. Right, and this time, these membranes of the thylakoids, there is no difference. There is one difference through here, the proton accumulation is towards the inside of the membrane, that is in the lumen. So in respiration, proton accumulation in the intermembrane space of the mitochondria, where electrons move through the ETS, electron transport system. So what exactly they have asked is ATP in terms of photosynthesis. So where do photon accumulation happens across the membrane during the time of photosynthesis? That is, photons accumulate in the intermembrane space of the mitochondrion. The next question, when tripalmitin is used as a respiratory substrate, the process consumes 145 molecules of oxygen and releases 1 or 2 molecules of carbon dioxide, then the RQ would be what? What is RQ? I hope you remember respiratory quotient. Excuse me. I hope RQ is nothing but respiratory quotient. You remember it. And there is one thing happening. Tripalmitin is used as a respiratory substrate. Then this tripalmitin is consumed. 145 molecules of oxygen is consumed and 1 or 2 molecules of carbon dioxide is released. What could be the respiratory quotient? Yes, B. So let's see whether it is B. Yes, the answer is B. So let's see some information regarding this respiratory quotient. So respiratory quotient is also called as respiratory ratio. It is nothing but the ratio of volume of carbon dioxide to the volume of oxygen consumed, or volume of carbon dioxide evolved to the volume of oxygen consumed during the respiration. So according to that, the value of respiratory quotient depends upon the type of respiratory substrate. What type of respiratory substrate is there here? Based on that, the respiratory quotient changes. So however, if it is, its value is 1 for carbohydrate, it is exactly 1 for carbohydrate. It is less than 1 for fats, because fat consumes more oxygen for respiration than carbohydrate. And here in the question, what we say is 145 molecules of oxygen is consumed and 1 or 2 molecules of carbon dioxide is released. So the substrate requires more oxygen for the oxidation, or more oxygen for their or more oxygen is consumed during this respiration process. Hence, if you do RQ value, if you put the value RQ is equal to 145 by 102, that is volume of oxygen consumed to the volume of carbon dioxide given out. Sorry, it should be on either the other one. So RQ is nothing but volume of carbon dioxide evolved to the volume of oxygen consumed. So here volume of carbon dioxide evolved is 102 and oxygen consumed is 145. When you divide this, you get the answer of nearly 0.7. So here the answer 0.7B is the right answer. I hope it is clear. So next, go for the next question. So in the diagram, there is stages of embryonic development has been represented. You have to identify the type of growth which is happening at M and N stage. Here M stage is given and the N stage is given in the picture. So this is the M stage, this is the N stage. You have to identify what is the M and the N labeling here. Is it M is a geometric phase and N is the arithmetic phase? Both M and N are arithmetic phase. M is an arithmetic phase and N is the geometric phase. Both M and N are geometric phases. So these phases can be seen in the embryonic development. So if you remember, the zygote divides further. So what we get is the answer. Yes. Yes, you are right. I have already shown the answer. It is A. So if you see the figure, the zygote is undergoing meiotic division. So in the initial stage of the M stage, every progeny cell retains its capacity to divide and form progeny cells. That is, no cell is undergoing differentiation. So it is dividing in 2n manner, right? So gradually what is happening? So as and when the division is happening, the number is changing. from 2 to 4, 4 to 8, 8 to 6, that is in arithmetic progression. What you see math, arithmetic progression. So it is dividing in the arithmetic progression. So this can be seen in the root and stem growth. So here basically the answer will be M is in arithmetic progression and N is in geometric progression, geometric phase. M is in geometric phase, N is in arithmetic phase. Why? Because your if you see here, they are dividing in 2n manner in the M phase, that is 2, 4, 6, 8. Correct. So that's why they say it as geometric phase, and then in the N phase, you can see the arithmetic progression. I hope the answer is clear. Then the 11th question, indigestion of fats in humans may be an indication of intestinal ulcers, under secretion of saliva, inflammation of liver, under secretion of amylopsin. So what could be the answer here? Indigestion of fats in humans may be an indication of intestinal ulcers, under secretion of saliva, inflammation of the liver, or under secretion of amylopsin. Yes, so I've got two answers here. So let's see whether that answer is right. So that is B. Your answer is B. That is the right answer, under secretion of saliva. Let's see why. Why it is B and why not D. So indigestion is often a sign of an underlying problem such as ulcers or gallbladder diseases. So here, indigestion of fats in humans may be an indication of inflammation of liver as well. Okay. So all these are, say, indigestion is often a sign of underlying problems such as ulcers, or it can be a gallbladder disease. It can be sometimes indication of inflammation of the liver here. So the question is, indigestion of fats in humans may be an indication of under secretion of saliva. If the fat has to be digested, the enzyme which comes in under the action is the salivary amylase. Salivary amylase. Is it right? Salivary amylase. So saliva is the one which helps in digestion of carbohydrate and the fat. And the first which comes under the indigestion is if there is under secretion of saliva. Yeah, Anvita, you say that, ma'am, but according to explanation, answer should be C. See, explanation says those are the signs, those are the indications. There will be gallbladder disease, there will be intestinal ulcers, there will be inflammation of liver. Everything happens. So, what exactly causes this indigestion of fats? That is under secretion of saliva is what we have to see. I hope it is clear. Next, we will go with the next. You have to choose the correct statement from the following. You have to choose the correct from the following. That is first statement is erythroblastosis fetalis may result when the fetus is Rh negative and the mother is Rh positive. The second statement is histamine, serotonin, and heparin are secreted by basophils. Then atherosclerosis is often referred as angina pectoris. And then person with blood group AB can donate, what is it? So person with blood group AB can donate blood to a person with blood group A. So what could be the right answer? I hope the voice is clear. Am I audible? The message is popping up. Am I clear, sir?
Yes, madam, you are audible. Hello. Hello. You're audible, madam.
So the answer, yes.
You can continue, madam. You're audible.
So right, the answer for this, which statement is correct? Erythroblastosis fetalis may result when fetus is Rh negative and the mother is Rh positive. Histamine, serotonin, and heparin are secreted by basophils. Atherosclerosis is often referred as an angina pectoris, and person with blood group AB can donate blood to the person with blood group AB only. So, A only. So, what will be the answer here? So, D. Anvita and Sneha says D. Let's see whether it is right. So, answer is B. The correct statement is B, they say. Let's see why. So erythroblastosis fetalis may result when fetus is Rh positive and the mother is Rh negative. So here the statement says fetus is Rh negative and mother is Rh positive. So that's the wrong statement there. Then atherosclerosis is often referred as arteriosclerosis, not angina pectoris. It is arteriosclerosis. And the person with blood group AB can donate blood to person with blood group AB only, but can receive blood from persons with blood group A, B, AB, and O. From everyone, they can accept the blood, they can receive the blood. Hence, they are called as universal receivers. But they can donate only to AB blood group. So hence, only B is the right statement among these four. The next question, in a blind spot of human eye, both cones and rods are absent. Only cones are absent. Both cones and rods are present. Only rods are absent. So what could be the right answer here? So in the blind spot of the human eye, so if you remember the structure of a human eye, there is a part in the human eye called as blind spot. So what is it? Is it because of both the cones and rods are absent, or only cones are absent in that spot, or both cones and rods are present, or only rods are present? What is the right thing regarding the blind spot in the eye? Anvita says B. Any other answers? Human eye. So at the end of the retina, you can see a small spot. We call it as blind spot. So what is the blind spot exactly? Both cones and rods are absent. Only cones are absent. Both cones and rods are present. Only rods are absent. What is it? So it is A, B, C, both, all three are there. All three answers I can see. So let's see the right answer here. So the answer is A. They say both cones and rods are absent. Let's see a little bit regarding it. So blind spot is a small portion of visual field of each eye. So if you see the structure of eye, so there will be a small portion in the structure of the eye. So this will be your visual field, correct? So in that portion, there will be a small spot where you can see that is where that corresponds to, that is within the retina, that there will be a small spot called as blind spot. So this blind spot will be deficient, or this will be the photoreceptor cells will be not there in this spot, or it will be deficient of this photoreceptor cells, that is both rod cells and cone cells. So hence no image formation takes place in this blind spot, as they are deficient of both cones and rods. So the next question, a boy after attaining sexual maturity shows muscular growth, growth of facial and axillary hair, aggressiveness, and low pitch of voice. These changes are attributed to dash hormone. So boy after attaining sexual maturity, he shows muscular growth, growth on facial and axillary hairs, then aggressiveness and low pitch voice. So what is this changes attributed to, or which hormone is responsible for all this? The question. So everybody is with the answer ready. B. So let's see. Yes, the answer is B, testosterone. So little bit we will see about what is this. So whatever the Leydig cells are there, or it is also called as interstitial cells, which are present in the intertubular space. So this produces a group of hormones called as androgens, and that mainly testosterone is secreted. So these androgens regulate the development and maturation and functioning of all the male sex organs. So testosterone is the one which is involved in increase in height, body and pubic hair growth, changes in the sexual characteristics, and aggressiveness and low pitch of voice. All these are controlled by testosterone. So the right answer here is B, testosterone. So let's go for the next question. So this is what we saw, 2019 questions of first PUC. So now we are going to see the 2018 questions of first PUC. The first question, 2018 paper, first question, in which of the following groups do the male and female gametes have independent free living existence? So in which of the following groups do male and female gametes have independent free living existence? Bryophytes, gymnosperms, bryophytes, pteridophytes, pteridophytes, gymnosperms, algae, and gymnosperm. What could be the answer? In which of the following groups do the male and female gametophytes have free living or the independent existence? Both male and female gametes are separate. In which of these? Bryophytes, gymnosperms, or bryophytes and pteridophytes, pteridophytes and gymnosperms, algae and gymnosperm. In which group do you see male and female gametophytes? They exist separately. I got the answer as A. Let's see. One is B, then any other? So let's see the answer. I have got A also as the answer as well as B. Let's see the answer. The answer is B. Why? Let's see here. So if you see algae, bryophytes, and pteridophytes have independent gametophytes. That is, they are independent. They have the independent gametophytes. Gymnosperms and angiosperms have dependent gametophytes. That is, in gymnosperms, male and female gametophyte do not have an independent free living existence and remain within the sporangia or retained within the sporophytes. This is what we can see in gymnosperms and angiosperms. And in all other things like algae, bryophytes, and pteridophytes, the gametophytes are independent in nature. Hence, the answer bryophytes and pteridophytes, that is B. The next question, you have to match the column 1 with column 2 by choosing the correct answer. Column 1 says actinodides, hemidactylites, Carcharodon, and Pteropus. On the other side, column 2, you have aves, Chondriactis, Mammalia, Reptilia, Osteactis. So what would be the right answer here? So you have to match column 1 with column 2 and tell me the right answer. So read the question properly and match it. Only Anvita has answered for this question, that is D. Any other answers? Neha says C. So let's see what is the right answer. It is D or C. The answer is D, right answer. That is actinodites are aves. Then Hemidactylus is reptilia. Carcharodon is Chondriactis and Pteropus is Mammalia. So that will be the right answer. That is the fourth one. So the next question, you have to see the picture and you have to choose the correct floral formula for this picture. So the picture is given here, floral diagram is given here. You have to choose the correct floral formula for this. I hope you remember the symbols and how to write the floral formula. Floral picture is given, you have to write the floral formula. I hope you are clear with what all these symbols given here. So what is the answer for third one? Anybody? Okay. Let's discuss then. I hope you don't know the answers. See, these questions have been appeared. This question has appeared in 2018 paper. So it's been asked there. So you have to be careful. You have to learn these kinds of questions as well. So let's see the answer. The answer is C, they say. So let's see a little bit more information regarding it. So in Brassiaceae, the flowers are actinomorphic. Actinomorphic means they are radially symmetric. Radially symmetric means when you cut half, you get the same equal half on either side. If you cut like this also, you'll get the same equal half. You cut like this also, you can get the same half. So hence they are called as actinomorphic or radially symmetric. Hence the symbol plus. Then they are bisexual, that is both male and female flowers are present. If they are bisexual, we represent it by the symbol a 0 with an arrow mark on its head and a plus mark on its tail. Sepals are 4 and they are dimerous, that is arranged in 2 whorls. Sepals we represent it by K and they are arranged in, So they are arranged in 2 whorls and they are like this. They are 2 are outside, 2 are inside. So we write it as 2 plus 2. And petals, there are 4 petals and they are arranged in single whorl. See here, petals are arranged in the single whorl. Petals are represented by C, that is 4. Then you can see the stamens. There are 6 stamens and they are tetradynamous, that is 2 lateral stamens and smaller, and 4 median stamens are larger. So if you see, these are the smaller stamens and these are the larger stamens where they are arranged. So where stamens or androgens, we call it as A, which is A, 2 plus 4. So 2 is smaller, 4 is the large one. Then carpels are 2. They are bicarpellary, syncarpus and ovary superior. So they are bicarpellary. There are 2 carpels and they are fused and they are superior in nature. So what you say, G2 and the bracket as they are fused. So this will be your floral formula, and according to that, C is the right answer. I hope it is clear to everyone, whatever the explanation is given. The next question, in which type of vascular bundles are xylem and phloem present at the same radius? In which type of vascular? There are different kinds of vascular bundles. So in that, which type of vascular bundles are xylem and phloem present at the same radius? Is it radial, closed, conjoint, exarch? In which type of vascular bundle you can see xylem and phloem are present at the same radius? Anita says it is A. So any other answers for this? So I'll repeat the question. In which type of vascular bundle you can see xylem and phloem present in the same radius? And is it radial, closed, conjoint, exarch? In which condition you can see this kind of arrangement? So let's see answer. Answer is C, conjoint, they say. Why the conjoint? When xylem and phloem are situated on the same radii of the vascular bundle, the arrangement is so called as conjoint, and the vascular bundle is called as conjoint vascular bundle. And such vascular bundles are common in stem and leaves of dicots and monocots. So here what they say, xylem and phloem, if they are situated in the same radius, so they are called as conjoint. They are called as conjoint, and the vascular bundles are called as conjoint vascular bundles. So such vascular bundles can be seen in the dicots and monocots, stems and leaves of dicots and monocots. Coming to conjunctive tissue is present between 1, 2, and 3. Where exactly you can see the conjunctive tissue? So is it between pericycle, endodermis, dicot, xylem, phloem, dicot root, palisade parenchyma, spongy parenchyma, dicot leaf, xylem, phloem, dicot stem? Where exactly you can see the conjunctive tissue? So where exactly you can see the conjunctive tissue? Option 4. Somebody says between xylem and phloem, the dicot stem. Any other answers for this? So where exactly you can see the conjunctive tissues? Is it between the pericycle endoderms in the dicot root, xylem, phloem in the dicot root, palisade parenchyma, spongy parenchyma in the dicot leaf, xylem, phloem in the dicot stem? So let's see the option, right option. The right option is B, between the xylem and phloem in the dicot root. So let's see some information regarding it. The parenchyma present between xylem and phloem in the vascular bundles of the root is called as conjunctiva tissues, conjunctive tissues. So in the parenchyma, whatever the parenchyma cells are there between the xylem and phloem is nothing but conjunctive tissue. So in the anatomy of dicot root, this parenchyma will be present between xylem and phloem. Okay, so this is what it is concerned in the formation of vascular cambium and storage of food and water. So this conjunctive tissue is meant for formation of vascular cambium and storage of food and water. So the exact answer is between xylem and phloem in the dicot root. So the type of epithelium found in the inner lining of PCT. I hope you remember what is PCT, proximal convoluted tubule. So which will be present in your excretory system. The type of epithelium found in the inner lining of PCT is squamous epithelium, cuboidal epithelium, glandular epithelium, ciliated epithelium. What could be the answer? So A, B, second option. What type of epithelium you can see in the PCT, that is proximal convoluted tubule. So let's see the answer. So is it A or B? Answer is B, the cuboidal epithelium. So let's see a little bit of that. So simple squamous epithelia, they are found in the capillaries, alveoli, glomerular, etc. So where the simple squamous epithelium is associated in filtration and diffusion. Simple squamous epithelia. And this type of epithelium found in the inner lining of PCT is known as cuboidal epithelium. The type of epithelium present in the inner lining of PCT is known as cuboidal epithelium. It consists of monolayer of epithelial cells, which appear square-shaped in cross-section. So when you take the cross-section, so you can see the cells like this square-shaped almost, the cuboidal shaped, and they are the one which will be lined or which will be present in the inner lining of the PCT. And what does it do? This epithelium, it helps, it is associated with, or it is present with, along with a large round centrally located nucleus as well. So this is cuboidal epithelium, which is present in the PCT, proximal convoluted tubule inner lining. So glandular epithelium will be present in major glands and intestinal lining, and ciliated epithelium will be present in the airways and uterus as well as fallopian tube. So the efferent ducts of testes and the ventricular system of the brain. Here all you can see ciliated epithelium, glandular epithelium in the glands, but cuboidal epithelium is present in the PCT, and squamous epithelium is present in alveoli, capillaries, and glomerulus. I hope which epithelium you can see in PCT is cuboidal. Is it clear? So we'll go for the next. So that is which the following graph shows the concentration of substrate on enzyme activity. What does the y-axis represent? Temperature, velocity of reaction, pH, or pressure. What does this y-axis represent in this picture? Yes. So what is that answer? Temperature, velocity of reaction. B option, velocity of reaction. Any other answers? What does the y-axis in the picture represents? That is regarding enzyme activity. I'll give the answers. The answer is B, velocity of reaction. So what is the reason? Let's see a little bit. So the substrate enzyme activity is represented by Michelis-Menten equation, which represents velocity on the y-axis. Velocity will be on the y-axis and substrate concentration will be on the x-axis. Okay, with the increase in the substrate concentration, the velocity of enzyme reaction is at first it increases and slowly it goes on. The reaction ultimately reaches a maximum velocity, which is not exceeded by any further rise in the concentration of substrate. And after these molecules, so there are no free enzyme molecules to find the additional substrate molecule. Hence, the y-axis represents the velocity of the reaction. 8th question. In the maize plant, carbon dioxide fixation occurs in both mesophyll and bundle sheath cells. The enzyme involved in these cells for the process respectively are rubisco and pep kinase and pepsin, rubisco and pep carboxylase, and rubisco. What could be the answer here? So we are talking about the 8th question, student. Option A, option C, option C. So you are saying rubisco and pep carboxylase. Sneha is telling rubisco and pep kinase. So let's see what is the right answer. The right answer is not even A or C, it is D, pep carboxylase and rubisco. So what is it? In C4 cycle, carbon dioxide fixation in mesophyll cells by pep carboxylase and synthesis of sugars in the bundle sheath cells by rubisco is what happens. For example, in maize plant, carbon dioxide fixation occurs in both mesophyll and bundle sheath cells, where the synthesis of sugar takes place in the bundle sheath cell with the help of rubisco. So pep carboxylase helps in fixing carbon dioxide and rubisco helps in the formation of sugar. So hence the answer, rubisco, pep carboxylase and rubisco. The following reaction, you have to identify X and Y respectively. So X means water, Y is acetyl-CoA, X is acetyl-CoA, Y is pyruvate dehydrogenase. X is pyruvate dehydrogenase, Y is acetyl-CoA, X is pyruvate dehydrogenase, and Y is oxalic acetic acid. Sorry, oxaloacetic acid. What could be the right answer? So you have to identify the picture and let me know what is the right answer here. Anita says 920. Sneha says it is C. Any other answer? Okay, D is given, C is given. So let's see the answer. The answer C is the right answer. That is pyruvate dehydrogenase and acetyl-CoA. So pyruvic acid combines with coenzyme A and NADH plus, forming pyruvate dehydrogenase, then carbon dioxide, NADH and H plus ion, along with the formation as acetyl-CoA. So what could be the reason or what could be the explanation? See here. The pyruvate, which is formed by the glycolysis, enters into mitochondrial matrix, which undergoes oxidative decarboxylation by a catalyst called pyruvate dehydrogenase. During this process, the two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid. This reaction is shown as follows. Pyruvic acid plus coenzyme A plus NADH plus, under the enzyme pyruvate dehydrogenase, forms acetyl-CoA plus carbon dioxide plus NADH plus H plus. So here, enzyme pyruvate dehydrogenase brings about carboxylation of pyruvic acid to form two molecules of acetic acid, which is further utilized in Krebs cycle. So X is pyruvate dehydrogenase and Y is acetyl-CoA. And the answer here is C, the pyruvate dehydrogenase and acetyl-CoA. Digestion of both starch and protein is carried out by the enzyme digestive juice, saliva, pancreatic juice, bile juice. Let's answer here. This is a very easy answer. I hope you remember. Digestion of both starch and proteins is carried out by the enzymes of gastric juice, saliva, bile juice, or pancreatic juice. What helps in digestion of starch and protein? Yes, so answer, let's see. So answer is D, pancreatic juice. What is the reason? Let's read out. So gastric juice, protein digestion takes place partially. Saliva, carbohydrate digestion partially. Bile juice has no enzymes. Then pancreatic juice has enzymes for digesting proteins, carbohydrates, and fats. The pancreatic enzyme includes amylase that digests the remaining complex carbohydrate and into monosaccharide. Trypsin, chymotrypsin, elastase all digest proteins. Lipase digests triglycerides into fatty acids and monoglycerides. Hence, without pancreas, starch, protein, and fat digestion cannot take place. I hope it is clear why we choose pancreatic juice. Which of the following factors are favorable for the formation of oxyhemoglobin in alveoli of the human beings? High POC, P carbon dioxide, low temperature, high H plus concentration, low PO2 concentration. What is it? Which of the following factors are favorable for the formation of oxyhemoglobin in the alveoli of human beings? So what is the answer? Which one is the favorable for formation of oxyhemoglobin? Which factors? Let's see the answer. Option C, option C, option C, option B. So let's see what is our right option. Option B is the correct option, that is low temperature. So let's see why. So normal temperature favors the binding of hemoglobin with oxygen. Okay, in normal temperature, oxygen binds or hemoglobin binds with oxygen. In the high temperature, it is responsible for dissociation of oxygen from the hemoglobin. As in when the temperature increases, oxygen from the hemoglobin is given out or dissociated. So in the alveoli, where there is high PO content, low PCO content, lesser hydrogen concentration and lower temperature, the factors are more favorable for the formation of oxyhemoglobin. It is lower temperature and the favorable conditions like high PO2 and low PCO2. These are all the factors which influences low temperature, high PO2, lower CO2, all this. And the factors which favors the formation of oxyhemoglobin is low, and that is the low temperature. The next question. Select the correct RH group of parent. So select the correct RH blood groups of parents whose child is affected with erythroblastosis fetalis. Both a father and mother are RH positive. Mother is RH positive, father is RH negative. Both father and mother are negative. Father positive, mother negative. What would be the answer? So, yes, Anita is telling me Any other answers? Yes, so let's see, you are giving D as an option. Let's see here. Yes, D is the right option. That is, father is positive and mother is negative. So let's learn few things about it. So when a mother is Rh negative, while the father is Rh positive, since the antigen producing gene is dominant, therefore expressed in the progeny, and so the fetus is Rh positive. Since the mother is Rh negative, the antigen present on the child's RBC can cause antibody reaction. Condition is called as erythroblastosis fetalis. That is the negative, and the child's positive, Rh positive, will have or will kill the each other, or the antigen will kill the antibodies. And thereby, the result is erythroblastosis fetalis. The hormone of fight, flight, and flight are thyroxine, oxytocin, thyroxine, melatonin, adrenaline, noradrenaline, gastrin, and secretin. What's the answer here? So the hormone which is for fright, fight, and flight are the hormones for fight, fright, and flight. Adrenaline and noradrenaline. People are telling C is the right option. So let's see the option there. Answer is C. That is adrenaline and noradrenaline. Correct. So the hormones of fright, fight, and flight is called adrenaline or epinephrine. It is secreted by adrenal cortex, and it helps the organism to offend, defend, or run away from the threatening situation. Adrenaline is a hormone produced by adrenaline glands during high stress or exciting situation. This adrenaline is produced. So this powerful hormone is a part of human body's acute stress responsive system. So it is called as fight or flight response. It works by stimulating the heart rate, contracting blood vessels, dilating air passages, all of which work to increase blood flow to the muscles and oxygen to the lungs. Additionally, it is a medical treatment of some potentially life-threatening condition during anaphylactic shock. I hope it is clear. Your answer C is right. Let's go for the next question. So now whatever the questions we discussed was KCET 2018. Now we are going to discuss about KCET 2017. So minimum to minimum 15 questions you will get from the first PUC syllabus, where you have to concentrate, and minimum 15 questions will be there. Okay, so I hope these questions will be helpful for you in one or the other way. So let's go for 2017 year paper of only first PU. So identify the order for the following. You have to identify the order from the following: carnivore, Muscidae, Insecta, Panthera. So what is the order you can see here? You have to identify the order here. Option B. So what's the answer here? What is the order here? Everyone is telling B option. Let's see. They say option A. let's see the reason why. So Carnivora is the order of eutherian mammals, which refer to the any meat-eating organisms. Means Carnivora is the order meant for or referred to as meat-eating organisms, or they belong to meat-eating organisms like dogs, cats, wolves, all that. And Muscidae is a family of flies. It's not the order. Muscidae is the family of flies found in the superfamily Muscodiae. The example is housefly, musca, etc. Then insecta are nothing but insects. These are the class. Insecta is a class of insects where hexapod invertebrates will be involved here. It is ants, bugs, etc. Hexapod means six-legged one. The Panthera is the genus here. It is not the order. Panthera is the genus. So here, Carnivora is the order. Muscidae is the family. Insecta is a class, and Panthera is the genus. So as they had asked for order, Carnivora will be the right answer. So you have to really careful with this. Which of the following option shows the characteristic of mycoplasma? Smallest living cells without cell wall survive with oxygen. Smallest living cells with cell wall survive with oxygen. Smallest living cell without cell wall survive without oxygen. Smallest living cell with cell wall survive without oxygen. So what could be the answer here regarding mycoplasma? So what are the characteristics shown by mycoplasma here? Anvita says it is A, smallest living cell without cell wall survive with oxygen. And Sushil says it is C, smallest cell without cell wall survive without oxygen. So what could be the answer? Yes, Sushmita also C, Harsha is also C. So you are telling C is the right answer. Let's see whether C or it is A. Yes, C is the right answer. That is smallest living cells without cell wall, and they survive without oxygen. So let's see a little bit information regarding it. So mycoplasma is the smallest cell which does not possess cell wall. The first thing is they don't possess cell wall as an outer covering, but they have cell membrane as the outer covering, and as they don't have cell wall, they don't get affected by the antibiotics. Okay, and they cause severe diseases to plants and animals. They can survive without oxygen and respire anaerobically. So they can survive, they can even survive without oxygen and respire anaerobically. Hence, they are the smallest living cells without cell wall, and they can survive without oxygen as well. So the next question, sorry, the answer is shown here. In which class of algae reproduces asexually by non-motile spores and sexually by non-motile gametes? Sorry, the answer is already shown. So let's discuss about it rather than asking us an answer. So the question says here, in which class of algae they reproduce asexually by non-motile spores and sexually by non-motile gametes? Answer is Rhodophyceae, right? Why Rhodophyceae? Why? What is that? We will see. So here, the algae which belong to group Rhodophyceae can reproduce sexually as well as asexually. So they Rhodophyceae group of algae, they reproduce both methods, sexually as well as asexually. So in say asexual method, they produce non-motile spores, and in the sexual method, they produce non-motile gametes, where the fusion takes place to form the embryo as well as new algal body. So then the next in the The next, that is for ACA, reproduction takes place both asexually and sexually, but here zoospores are formed in asexual reproduction, and sexual reproduction takes place both by isogamy and anisogamy method. In Chlorophyceae, sexual reproduction occurs isogamy, anisogamy, and oogamy process. And in Cyanophaceae, that is blue-green algae, reproduce both vegetatively and asexual means. That is vegetatively through fission and asexual means as well. So the algae which shows this kind of thing is Rhodophyceae. The fourth question, which of the following plants produce zygomorphic flowers? Which of the following plants produce zygomorphic flowers? Hibiscus, canna, gulmohar, mustard. What is the answer here? So, which of the following plants produce zygomorphic flowers? Is it hibiscus, canna, gulmohar, or mustard? What could be the answer here? Sushil says it is C. Any other answer for this? Zygomorphic flowers. Which plant produces zygomorphic flowers? So is it hibiscus, canna, gulmohar, mustard? Anvita says it is A. Sushil says it is C. What is the right answer? It is A or C. Hibiscus or gulmohar? Or it is canna or mustard? Which one? Right. So let's see what is the right answer. So it is answer C. C is the right answer. Sushil is right. C. Why C? Why not A? Let's see here. So if you see, zygomorphic flowers have the symmetry in which flowers can be divided into two halves only through the division in one plane. Means, as we said, actinomorphic flowers means we can cut either this way or this way, right? So zygomorphic flowers are the flowers where you can cut either this way or the vertical position, vertically or horizontally. Actinomorphic means both vertically and horizontally, but in zygomorphic, either this or this, either vertically or horizontally, right? A flower which cut in two equal halves through any vertical plane passing through the axis, then its symmetry remains undisturbed. Such flowers are called as actinomorphic flowers. Okay, so if you see, if a flower is cut into two equal halves and on either side the symmetry remains undisturbed, they are called as actinomorphic. As I said, this one. These are actinomorphic. Any of the symmetry, if you cut, you'll get same equal halves and they are not disturbed. They'll be same, right? Sorry. And the flower of the plant gulmohar are zygomorphic in nature. The flowers of the plant gulmohar are zygomorphic in nature. Canna, mustard, hibiscus have the flowers of actinomorphic. So canna, hibiscus and mustard, if you see, they are actinomorphic in nature. They can give symmetry in either ways, but gulmohar is the only one which is zygomorphic in nature. It has to cut in one particular plane. Fifth question, the secondary wall material suberin is deposited on the walls of. The secondary wall material suberin is deposited on the walls of pericycle of the stem, endodermis of the root, phloem of stem and endodermis of root, epidermis of stem and endodermis of root, phloem and phloderm. Where exactly suberin is deposited? So where you can see the suberin deposition here. The secondary wall material, that is suberin, is deposited on the walls of pericycle of stem and endodermis of root, phloem of stem and endodermis of root, epidermis of stem and endodermis of root, phelogen and phylloderm, where exactly you can see the suberin deposition. So is it the answer C? The Sneha and Harsha is telling answer C. Any other answer for this? So let's see the right answer. The right answer is B, not C. That is phellem of stem and endodermis of root. Let's see why. So suberin is a fatty material which is deposited on certain cells of the plants. So the material is deposited on the phellem cells in the stem and the endodermal cells of the root. On the endodermis, the suberin deposition results in the formation of Casparian strips, which prevent the flow of water. So why they are present in the endodermal region of the roots? Because they prevent the flow of water. So the material is deposited on the phellem cells in the stem and the endodermis of the root, and the answer will be B. I hope the explanation is clear. The next is the type of epithelium found in the fallopian tube which functions to move the particles or mucus in specific direction is squamous epithelium, cuboidal epithelium, ciliated epithelium, columnar epithelium. So the type of epithelium which you see in the fallopian tube, which helps the particles or mucus to move in specific direction. What epithelium is it? Squamous, cuboidal, ciliated, or columnar? So it is ciliated, you say. Option C, ciliated. So what is the option? Any other answers for this? C option. So let's see, everyone is giving C as an option. Yes, the right answer is C. So let's see what is the explanation for this. The internal lining of the tube-like structures, the interlining of the tube-like structure, and so there will be tube-like structures which possess ciliated epithelium. So the internal lining of the fallopian tube, there will be ciliated epithelium present. So they bear short hair-like projections. They are called as cilia, which beats and allows the movement of particles, which allows the movement of particles. So their function is to move the particles of mucus in specific direction over the epithelium organs like bronchioles and fallopian tubes. So this ciliated epithelium usually present in the bronchioles and fallopian tubes. Their main function is to move the particles and the mucus in one particular direction, as they have cilia or the hair-like projections. The seventh question, which of the following is not included under the endomembrane system? Which of the following is not included under the endomembrane system? Endoplasmic reticulum, mitochondria, lysosomes, vacuoles. So what is not included in the endomembrane system? So now we are in seventh question. We are talking about the seventh question of 2017 paper. So which of the following is not included under endomembrane system? Is it endoplasmic reticulum, mitochondria, lysosomes, or vacuole? So Sneha says it is B. Any other answer for this? C. Which one of the following is not included under endomembrane system? Which of these following organelles are not included in the endomembrane system? So let's see option A, B, C, D, all options you have given. Let's see the right option here. Option B is the right option. That is mitochondria. Why mitochondria? What is it? Let's see. The endomembrane system is composed of membranous structures in the cell. That is which can have membranes. So the common structure which are included are lysosomes, vesicles, vacuoles, cell membrane, endoplasmic reticulum, nuclear membrane, etc. Mitochondria, peroxisomes, chloroplast are not included under endomembrane system. So their membranous structures are not included in this. The endomembrane system doesn't come. So these three organelles, that is mitochondria, peroxisomes, and chloroplast, doesn't come in endomembrane system. And all the other remaining, they are composed of membranes, and they all come under this endomembrane system. So mitochondria is the one which doesn't include under endomembrane system. I hope it is clear. The next question. Identify the major site of biosynthesis of lipids. Identify the major site of biosynthesis of lipids. Golgi apparatus, mitochondria, endoplasmic reticulum, that is smooth endoplasmic reticulum, SER, and rough endoplasmic reticulum, RER. So you have to identify the major site of biosynthesis of lipids. That is, where do the lipid biosynthesis takes place? Is it in Golgi apparatus, mitochondria, smooth endoplasmic reticulum, or rough endoplasmic reticulum? So everybody is saying C. Let's see. Yes, it is smooth endoplasmic reticulum. Okay, why? Okay, Disha, why it is not mitochondria? Let's see. So if you see endoplasmic reticulum is that organelle which you see in the eukaryotic organism that forms an interconnection between the cell membrane and the nucleus, right? So here it will be the nucleus. So here you can see the endoplasmic reticulum formation. So where few endoplasmic reticulum will be lined with ribosomes and few will be not lined with ribosomes. The endoplasmic reticulum which is lined with ribosome, we call it as rough endoplasmic reticulum or RER. Those endoplasmic reticulum which are not lined, we call it as smooth endoplasmic reticulum, that is SER. So if it is involved with ribosomes, ribosomes are meant for protein synthesis. They are called as protein factories. So if endoplasmic reticulum is associated with the ribosomes, they are involved in biosynthesis of proteins. And as the endoplasmic reticulum is lacking ribosomes, they don't have ribosomes, they get involved in the biosynthesis of lipids. So here you can see I have highlighted active smooth endoplasmic reticulum lacks ribosomes and functions in lipid metabolism and biosynthesis and carbohydrate metabolism, etc. So the right answer C. Which of the following is not a characteristic of facilitated transport? Uphill transport, highly selective transport, requires special membrane proteins, then transport saturates. So what could be the answer? What is not the characteristic of facilitated transport? I hope you all remember about the facilitated transport. So what is not the characteristic of facilitated transport? Nobody knows the answer. So she gave A as the option. Any other option here? A. Everyone says it is uphill transport. So let's see what is the right answer. Right, it is uphill transport. What exactly is that uphill transport? What all the others? Let's see. So facilitated transport is nothing but a selective transport of ions or molecules through special membrane called as channel proteins. So in the membranes, in the cell membrane, you have channel proteins where they are meant for selectively transporting these ions and molecules. Right, so the transport depends upon the concentration gradient or the saturation limit. And the movement is in accordance with the concentration gradient itself. So hence it is a downhill transport. It is not the uphill transport. And the transport proteins provide high selectively and transport only certain molecules. And the uphill transport is an example of active transport in which there is transport opposite to the concentration gradient. So you have to keep in mind uphill transport means the transportation happens opposite to concentration gradient. But here in the facilitated transport system, if the concentration gradient is more here, it moves to the opposite direction wherever it is less. Right. So the transport system is a downhill transport. It is not the uphill. And hence, as I said, they are selectively, highly selective. They select only certain ions and molecules to pass through, and they are associated with special membrane proteins, and there is a transport saturation also. So hence, these three are the characteristic of facilitated transport. Uphill is not the characteristic. It should be downhill. So identify the element whose deficiency causes both necrosis and chlorosis. Identify the element whose deficiency causes necrosis and chlorosis. So you have to find the elements which is responsible for necrosis and chlorosis here. The elements whose deficiency causes necrosis and chlorosis. Which are those elements? So everyone says it is A. So the element which causes necrosis and chlorosis. So let's see what is the right answer and what is necrosis and chlorosis as well. Yes, your answers is right. Magnesium and potassium are the elements whose deficiency causes necrosis and chlorosis. So let's see what is it. The plant requires the element for the growth and developmental processes. Okay, so the deficiency of these elements causes adverse effect on the growth of the plant. So the deficiency of this magnesium and potassium is very much required for the proper development. That is, magnesium and potassium are required for the proper growth and development of the plant. If there is deficiency of this, it causes necrosis and chlorosis. Necrosis is the death of all the living tissues, and chlorosis is decolorization, that is depigmentation of leaves. So in the leaves here and there, you can see the patches, yellow patches or white patches formation. So where decoloration would have happened, that is called chlorosis, and necrosis is death of the living tissue. Okay, so that is due to the deficiency of elements magnesium and potassium. So the outcome of Calvin cycle is, the outcome of Calvin cycle include what? So the outcome of Calvin cycle results in what? So your answer should be what? You have to find the right option here. 6 carbon dioxide, 18 ATP, 12 NADPH, or 1 glucose, 18 ATP, 12 NADPH, 6 carbon dioxide, 18 ATP, 12 NADPH, or 1 glucose, 18 ADP, 12 NADP. So you have to remember, hope you remember the Kelvin cycle. So if you remember the Kelvin cycle, you can answer. What could be the answer here? So if you have given B as an option, so let's see whether it is the right option or not. No, option D is the right option. Why D? Let's see. So I'll give you the picture as well. So if you see, this is the Kelvin cycle, right? So carbon dioxide fixation occurs during this Kelvin cycle, the carbon dioxide from the atmosphere. So the fixation of this happens where there are 3 stages in the process of Kelvin cycle. One is carboxylation, the other one is reduction, the third one is regeneration. So one turn of the Kelvin cycle fixes 1 molecule of carbon dioxide. You get 1 molecule of glucose and 18 ADP and 12 NADP. Right? I hope you got this. So this cycle, once if it happens in 3 stages, that is carboxylation, reduction, and regeneration. And during this process, 1 molecule of carbon dioxide will be fixed to get 1 molecule of glucose, 18 ADPs, and 12 NADP. I hope you got the answer clear. So the next question, the number of ATP molecules utilized for the breakdown of 1 molecule of glucose during glycolysis is. So the number of ATP molecules utilized for the breakdown of 1 molecule of glucose during glycolysis is 4, 2, 6, 8. So how many ATPs are required to break down a glucose molecule during the process of glycolysis? So how many ATP molecules are required? Any answers for this? He has given C. C is the option. How many ATP molecules are required or utilized during the glycolysis process so that the glucose is broken down? And this Neha has given the option. Let's see what is the right option. Option B is the right option, that is 2. So let's see what is it. So glycolysis is a process in which partial oxidation of glucose molecule is carried out. Glycolysis is the process in which partial oxidation of glucose takes place, where glucose molecule will be broken down to give 2 molecules of pyruvate, if you remember. So where 4 ATPs are produced, 2 are used up back, and the net yield will be 2 ATP given out. So the number of ATP molecules utilized during the breakdown of 1 molecule of glucose is 2 ATPs. I hope the answer is clear. So now match. You have to match the column 1 with column 2. So just check, the column 1 has enzyme and the column 2 has the functions of that enzyme. So you have to match the correct enzyme with their function. So the column 1 has enterokinase, renin, amylase, lipase. Column 2 has digest milk protein, digest carbohydrate, activates triglycerides, then acts on nucleic acids, breakdown of fats. What is the function of these enzymes exactly? Sneha says A. Any other answer? Asha says C. A, C. So there is a confusion between A and C. So any other answer, any other option to be given? Let's see the right answer here. The right answer is A, that is enterokinase activates triglycerides. The renin digests milk proteins. Amylase digests carbohydrate, and lipase is breakdown of fats. So the right option is A. Yes. So the next question, the volume of air inspired and expired by a healthy man per minute is? The volume of air inspired and expired by the healthy man per minute is 1000 volume to 1100 volume, 2500 ml to 3000 ml, 6000 ml to 8000 ml, 400 ml to 500 ml. So what is the volume of inspired air to expired air? That is, when you breathe in, how much volume of air you take in, and when you breathe out, how much volume of air you pour out or you give out. How much is that? You have to let me know the options here, A, B, C, D. Option A says 1000 ml to 1100 ml, 2500 ml to 3000 ml, 6000 ml to 8000 ml, or 400 ml to 500 ml. What is the option? So is it C, D, which one? C option. So let's see what is the right option here. C is the right option, that is 6000 ml to 8000 ml. Let us see a little bit regarding it. So if you remember, tidal volume is what we have learned in the first PUC. Tidal volume is the lung volume, which represents the normal volume of displaced between the normal inhalation and exhalation. When we normally inhale and exhale, whatever the amount of air we take in and give out, that is called as tidal volume. So when extra effort is not applied, general, normal breathing. So in a healthy man, young human adult, the tidal volume is approximately 500 ml per inspiration. That is, the breathing rate of the normal person is 12 to 15 breaths per minute. So for one inspiration, 500 ml. So he will take 12 to 15 breaths per minute. So altogether, he inhales 500 into 12 is 6000, 15 into 500 is 8000. So altogether, he inhales 6000 ml, exhales 8000 ml per minute. I hope this calculation is clear. So the next question, in the following diagrammatic representation of the standard ECG, the T represents here what? This T represents what? Depolarization of atria, depolarization of ventricles, repolarization of atria, or polarization of ventricles. What is the right option here? What is T represents in the picture? D option, the repolarization of ventricles. So she says D. Harsha also says D. Any other answer? D. So let's see what is the answer. Right, it is repolarization of ventricles. So let's see a little bit regarding it. What is it? So,
Madam, no break.
Sir, this is the last one, I guess.
Okay.
My session also completes.
Okay, ma'am.
Each peak in this ECG is identified as the letter P, Q, or S until T corresponding, whatever you can see. So where I'll just wind up. T wave represents the return of ventricles from the excited to the normal, that is repolarization. P represents electrical excitation. Q, R, S, this big thing represents a depolarization of ventricles, and T represents repolarization of the ventricles. And yes, I hope this is the last question. There are only 2 questions left. So I'll send these questions to sir and you can get the answer.
Yeah, you can send this material, madam. I'll share it to the students.
Yeah. Thank you very much.
Thank you, ma'am. Students, now you can go for break for 15 minutes. 6:05 sharp class will start. Chemistry, join positively at 6:05.
Dayanand Sagar University's College of Allied Health Sciences in Harohaldi, Bangalore. We're changing the game in medical diagnostics and treatment. Our innovative program combines theoretical knowledge with hands-on clinical training at our 300-bed tertiary care hospital, soon to expand to 1,350 beds in 3 years. Our cutting-edge infrastructure includes an advanced skill lab and simulation center to prepare you for global healthcare challenges. Plus, with regular workshops and conferences, you'll stay up to date on industry trends. Our strong curriculum is aided by technological advancements, and you'll have opportunities for innovative research at our Central Research and Innovation Wing. Gain real-life experience with 1 year of industrial exposure. Experience academic excellence in a global environment at Dayanand Sagar University. Live the dream. Welcome to the School of Engineering at Dayanand Sagar University. Main campus, Harohaldi, Road, Bangalore. A place that values research, innovation, and creativity. Our industry-aligned curriculum offers a blend of theoretical knowledge, practical experience, academic projects, and internships for a rewarding career in engineering. With an interdisciplinary approach, you can explore your interests by selecting open electives. The on-campus innovation labs, led by professors of practice, provide immersive learning and expertise to find the best solutions to real industry problems and give a different dimension to the students' mini and major academic projects. Our Center for Entrepreneurship supports startup opportunities and provides a perfect platform to launch your next big idea into successful ventures. We also prepare you for advanced studies in the latest futuristic technologies. Our BTech programs offer the perfect combination of academic rigor and functional experience in this ever-evolving technological landscape of the future. Experience a whole new world of academic excellence in a global environment. Dayanand Sagar University. Live the dream. Nestled within the beautiful and peaceful setting of Dayanand Sagar University's Harohaldi campus is the prestigious College of Nursing, where you embark on a transformative journey as we flawlessly fuse academic expertise with practical skills through our advanced curriculum. Renowned for our focus on innovation and advancement, our experienced faculty members are at the forefront of the nursing field, constantly incorporating the latest advancements in technology and research into the curriculum. Our pioneering facilities, including simulation labs and clinical training rooms, provide an immersive, real-world healthcare environment. The Harohaldi campus is a modern and supportive community, complete with all necessary amenities, and provides the perfect, serene environment for students to focus on their studies and achieve their goals. Join our vibrant community and prepare for a successful career in nursing. At Daya Sagar University's Harohaldi campus, the College of Physiotherapy is dedicated to providing students with a top-notch education in the field. Curriculum is designed to give students a comprehensive understanding of the human body and its functions, along with experiential training in the latest physiotherapy techniques. What sets our department apart is our ultra-modern facilities, including sophisticated classrooms and clinical training rooms. Our students have access to the latest technology and equipment, allowing them to hone their skills and knowledge to the highest degree. The faculty at the College of Physiotherapy is comprised of experienced professionals who are dedicated to their students' success. With our modern amenities, the supportive community, Daya Sagar University's College of Physiotherapy is the ideal place for aspiring physiotherapists to begin their journey. Welcome to the School of Engineering at Daya Sagar University, main campus, Harohaldi, Kanakpura Road, Bangalore. A place that values research, innovation, and creativity. Our industry-aligned curriculum offers a blend of theoretical knowledge, practical experience, academic projects, and internships for a rewarding career in engineering. With an interdisciplinary approach, you can explore your interests by selecting open electives. The on-campus innovation labs, led by professors of practice, provide immersive learning and expertise to find the best solutions to real industry problems and give a different dimension to the students' mini and major academic projects. Our Center for Entrepreneurship supports startup opportunities and provides a perfect platform to launch your next big idea into successful ventures. We also prepare you for advanced studies in the latest futuristic technologies. Our BTech programs offer the perfect combination of academic rigor and functional experience in this ever-evolving technological landscape of the future. Experience a whole new world of academic excellence in a global environment. Daya Sagar University. Live the dream. At Daya Sagar University's Harohaldi campus, The College of Physiotherapy is dedicated to providing students with a top-notch education in the field. Curriculum is designed to give students a comprehensive understanding of the human body and its functions, along with experiential training in the latest physiotherapy techniques. What sets our department apart is our ultra-modern facilities, including sophisticated classrooms and clinical training rooms. Our students have access to the latest technology and equipment, allowing them to hone their skills and knowledge to the highest degree. The faculty at the College of Physiotherapy is comprised of experienced professionals who are dedicated to their students' success. With our modern amenities and a supportive community, Dayanand Sagar University's College of Physiotherapy is the ideal place for aspiring physiotherapists to begin their journey.
Dear students, another just 2 minutes left. Ask your friends to join back. You to join back, students. All of you. Chemistry, very important class. Again, the revision. Dear students, join back. Ask your friends to join back fast. Enter just 1 minute. Dear students, I'll hand over the session to Ravi Shankar, sir. Sir, over to you. Your screen is visible. You can take over, sir. Sir? Am I audible, sir?
Murli sir, is it connected now?
Audible, sir.
Video is coming.
A video may not require, sir. Your voice is sufficient and your material is presentation is audible.
Okay, sir. No problem. Thank you, sir.
Okay.
Dear students, welcome back to our last session in chemistry today. Okay.
Last class they'll put queries in Q&A box, sir.
No problem. Let them. Okay. So we'll start one more mock exam today. Last class we have done one. Today also we'll do it. If possible, I don't know whether time will permit or not. If possible, the third also. Okay. We'll start first one. So what is this question? With respect to conformers of ethane, which of the following statement is true? What may be the answer? Anybody? Sujal, tell me what may be the answer? Yeah, please give your answer, all of you. What may be the option? Quickly, all of you. Charmila C. Unknown A. All of you, just give me option, whatever it may be. The first option says bond angle remains same, but bond length changes. In second, bond angle changes, but bond length remains same. C, bond angle and bond length change. Both bond angles and bond remains same. What may be the option? Anybody? So what are these conformers? What are these conformers? I will take other color. So what are these conformers? There's another name for conformers. Anybody knows it? They call it as conformers, or we can also call it as rotamers. We can also call it as rotamers. So conformers are rotamers. What are these one? They differ in spatial arrangement. So they differ in spatial arrangement where around carbon-carbon single bond. And how to represent? We can represent by using Sar's projection formula or Newman projection formula, but very frequently we use Newman projection formula. That's what we use. So for example, if I take ethane as an example, what is ethane? CH3 is the ethane. And imagine you're observing the molecule standing here. You're standing here and you're observing it from this angle. Then this is known as first carbon. We write it as dot. And the hydrogen attached to it, we write it in a long bond, in a bond angle of 120 degrees. The second carbon is written as a ring. And the hydrogen attached to it is written as a short bond. If the atoms of second and the first, if they're away from each other, we call it as what? Staggered. We call it as staggered. And we know that it is highly stable because repulsion is less, energy is less. But the angle between these first carbon and the second carbon hydrogen, we call it as a dihedral angle. It is around 60 degrees. So in conformers, what is happening is that in conformers, please remember the bond length remains same, sir. It will not change. It remains same. The bond angle also remains same. Only what changes? This 60 degree I told you, dihedral angle we call it as. We call it as what? Dihedral angle. That will change, sir. Dihedral angle will change, but angle remains same. What is the bond angle? Bond angle is the angle between two bond pair. So option is the 3 is the right statement, sir. What will change? Only dihedral angle will change. Angle will change. In a staggered, it's 60 degree, whereas in eclipsed, sir, in eclipsed, it is 0 degree. 0 degree. And between eclipsed and the staggered, there will be thousands of conformers will be there. The bond angle may vary from 0 to 60. Only the dihedral angle changes, but the bond angle and bond length remains same. Next one. So what may be the option? It's the third option. So it remains same means, sorry, that option is 4. So what is happening? Only diagonal angle will change. The only that will change. All others will remain same. Option is 3. Sorry, option is 4. Next one. Of the following, which is the product formed when cyclohexanone undergoes aldol condensation followed by ET? What may be the option? He only says aldol condensation. So what is aldol condensation? Anybody? So what is aldol condensation? Aldehyde having alpha hydrogen atoms reacts with dilute alkali. Alkali means any base like NaOH, KOH, barium hydroxide. They form what? They form a compound called as aldol. And this upon heating loses water molecule to form alpha beta unsaturated aldehyde. Unsaturated. This kind of reaction we call it as a aldol condensation. So how this reaction takes place? He only says cyclohexanone. Cyclohexanone means ketone. So here is not aldehyde, is a ketone. And you will not get aldol, you get a ketol. You will not get unsaturated aldehyde, but you get unsaturated ketone. That's it. That's the only difference. So that is the only difference. So instead of aldehyde, you get ketones. Instead of aldol, you get ketol. Instead of unsaturated aldehyde, you get ketone. When you say unsaturated, it needs to have a double bond. That means option 1 is not possible, option 3 is not. It needs to have a one ketone group, not two ketone group. Look at it, it's having two ketone groups. So option 4 is also not. So only option is second is the answer. So how this happens? What is cyclohexanone? Cyclohexanone is having, sorry, not this one, is having a ketone group. Is it not? When this reacts with alkali, what is alkali? Base. What is base? It is having OH minus ion. And the CH2 here will give one H plus 2 OH minus ion and it becomes water. So what you get? You get a CH minus ion. This we call it as a enolate ion. So this is the first step, enolate ion. This enolate ion will attack another cyclohexanone. So it will attack another cyclohexanone molecule. Where it attacks this particular cyclohexanone, this negative charge, no, this carbon is having positive charge. And this minus will attack this carbon atom, carbonyl carbon atom. Carbonyl. So what will happen? I will write it again. So this is the cyclohexanone. This will attack another cyclohexanone. I will write the cyclohexanone. And next to that carbonyl carbon is attached to it is a ketone group. That is, it becomes O. Ultimately, it accepts a proton from the solution, becomes OH. OH. What you are having here? A ketone is there. Since this compound is having a ketone as well as alcohol, it is called as ketol. So this is our ketol. Then what you're doing, you're eating it. You should lose water molecule. Yes, sir, let us observe what will happen. this OH, and here what you're having? You're having a CH. This OH and the CH, they combine with each other to form water molecule. So what is remaining now? So what is remaining is a cyclohexane ring with a ketone that remains as it is. In that C, sir, H has gone. And the OH is also is removed. So this is cyclohexane ring. When that OH is removed, you get double bond. So a ketone with a double bond. Yes, sir. Option second is right. Option second is right. Got it, all of you? If yes, please put it yes. Sushil, it's not A option. It is a second option. Yes. Next one. A concentration of Ag plus ion in a saturated solution of, what is this? Silver oxalate. Silver oxalate is 2.2 into 10 to the power minus 4 mole per liter. The solid product of silver oxalate is, how much it is? What is the solid product of silver oxalate? Give me option, all of you. Let me see what are the right answer. What will be the option? We know that this silver oxalate, I'll take another ink, red ink I will take. Ag2 he has given. It is silver oxalate. Will dissociate as, it gives two ion. One is Ag plus ion. How many? So it gives two Ag plus ion. And it gives oxalate ion. What is oxalate? C2O4, 2 minus oxalate. So remember this one. So this is the equation. What is the solid product? We know that solid product in a saturated solution is nothing but is a product of molar concentration of ions. Which is the ions? Sir, H plus ion. How many moles is there? 2 moles. Raise it to the power. Then molar concentration of oxalate ion. What is oxalate ion? C2O4, sir, minus 2. And we know the silver ion concentration they are given. How much they are given? Sir, Ag plus concentration, what they have given is 2.2 into 10 to the power of minus 4. Minus 4 they are given. So they're asking what? So they're asking solid product of silver oxalate. Silver oxalate. What may be the option then? Anybody got it? Give me option. Sir, option 3, D, B, Sridhar, D, Inchara, option 4. Anybody else? So how the equation is? So the equation is like this. If you take S mole of silver oxalate, it gives you 2 moles of Ag plus ion, 2 moles of Ag plus ion, and S mole of oxalate ion. Oxalate ion. So the concentration of oxalate, so concentration of oxalate becomes what? Ksp divided by concentration of Ag plus. Ag plus. And this can be written as what? They are given 2.2 into 10 to the power of minus 4. Minus 4. If you take this one, what we get? Sir, half of it. There is 1.1 into 10 to the power of minus 4. Minus 4. So we get a concentration of oxalate ion. Once we know the concentration of oxalate ion, we can substitute it here. Ksp is equal to, so 2.2 into 10 to the power of minus 4 to the power of 2. And 1.1 into 10 to the power of minus 4. How much it will be? So it will be 5.3 into 10 to the power of minus 12. So option will be, sir, fourth one is the answer. Next one. Mixture of chloroxylenol and terpenol is a. Which chapter? So this is from chemistry, nevertheless. And we know that there we have discussed about antiseptic. What are the antiseptic? Sir, Dettol is an antiseptic. Celol is an antiseptic. Then what are the other antiseptics? We have discussed about hydrogen peroxide. Then iodoform is antiseptic. Tincture, we call it as. Sir, all these are antiseptic. And here, Dettol is a mixture of what? Sir, chloroxylenol and terpenol. That means it is antiseptic, sir. Antiseptic means option is 2, sir. Next one. The vitamin that helps in clotting of blood is. Which are the vitamins will help in clotting of blood? And we know that in a water-soluble vitamins, there are mainly 2 things. Vitamin B and vitamin C. And whereas in fat-soluble, sir, we have 4 vitamins, A, D, E, and K. A, D, E, and K. You know that A is for, very important for eyes. D is for, sir, bones. E is for muscles. And, sir, K is for, this causes, sir, delayed blood clotting. If it is not there, it causes delayed blood clotting. So for clotting, what is important? Sir, E is important. E is important. So vitamin K is important. What is the name of K? Piloquinone. Piloquinone. That is very important, sir. Very important. So option is B, sir. Option is. Next one. The polymer containing a 5-methylene group is. What is a methylene group? The CH2 group is known as methylene. Which polymer contains a 5-methylene group? Sir, Dacron cannot be. Why? Dacron is made up of what? Sir, glycol and a terephthalic acid. So it cannot be. Sir, bakelite. Sir, bakelite is made up of what? Phenol plus formaldehyde. So it cannot be. There you will not get these methylene groups. So only option is nylon. Yeah, but in nylon 6,6, you know that it is made up of what? Hexamethylene diamine. But he says 5. In nylon 6, what we are having is a hexamethylene diamine. So that can also be not possible. So only option is nylon 6. And nylon 6, you know that it is made up of what? Caprolactam. Caprolactam. So what is this caprolactam? It is having 6 carbon atom. One is CO, other is NH group. So how many groups are required now? 5. Because nylon 6 means it is made up of 6 carbon atom. So again, I'll put CH2 here and CH2 here. Another 2 is required. I put CH2 here and CH2. So now look at here. How many CH2 groups are there? So there are totally 5 methylene group is there. Where? In caprolactam, which makes nylon 6. So option is Sir D. Next one. So cis-1,4 polyisoprene is called as what? We know that it is a rubber. What is rubber? So rubber is made up of cis-isoprene. And how these isoprene units are joined to each other? They're joined to each other through this 1,4 linkage. That's why they call it as a cis-1,4 polyisoprene. So it's a natural rubber. Natural rubber means option is B, sir. Option is B. Next one. Which cleaning agent gets precipitated in hot water? Means which can form a scum in hot water. What is a scum? Scum is a calcium salt. Remember this one. Of what? Fatty acid. Fatty acid. So where do you get this calcium salt of fatty acid? So where we get it? Sir, hard water will have a calcium ions. Hard water will have 2 ions mainly. One is calcium, other is magnesium ions. And our soap is made up of what? So soap is made up of a sodium salt of fatty acid. Fatty acid. Sodium salts are fatty acid. Then which is a fatty acid, sir? This one, sodium stearate is a fatty acid. So then answer is D, sir. Because only soap forms a scum. When soap reacts with hard water, so it forms a scum. So detergents, no. Detergents can be used in any water. With hard water also, they will not give any precipitate, no precipitate. Then which are the detergents? Sir, all option A, B, C, all these are detergents. Detergents. So cetyl, sir, this is a cationic detergent. Whereas B and C is there, no sir, these are the anionic detergents. So option has to be D, sir. Option has to be D. This is how the reaction takes place. To form a calcium stearate is a insoluble precipitate. We call it as a scum. Antihistamine among the following is. So what is antihistamine? Means they're the allergic drugs. Allergic drugs are usually called as antihistamine, remember. But remember, even antacid is also called as antihistamine. Antacid is also antihistamine, but very popular is that if anybody says antihistamine, it is mainly allergic drug. So what is amoxicillin? Sir, amoxicillin is an antibiotic. So this is antibiotic. Chloroxyrenol, sir, is an antibiotic. Morphine, sir, is a painkiller. So all this cannot be the answer. Over. You can remove the odd man out. So what should be the option? Bromopeniramine. So this is an allergic drug. What is allergic drug? Which blocks histamine receptor. That's why they're called as antihistamine. Next one. Create the correct intermediate and product in the following reaction. First, no, you have to, I told you, you have to understand which chapter. Look at here. There's a triple bond. That means, so it's alkyne. Alkyne. Alkyne means which chapter? Sir, this is a hydrocarbon chapter. And also it belongs to aldehydes and ketones also. Why, sir? The same reaction is used to prepare aldehydes and ketones. So it belongs to two chapters. One is hydrocarbon, another is aldehydes. What is this reaction? You are adding water. That means it's hydration. So it's a hydration of alkyne. And I'll give you one shortcut. Remember this one. What is that? So whenever you add water to ethane, what is ethane? Acetylene. Whenever you add water to acetylene, always remember you end up getting only aldehyde, acetaldehyde. Any other alkyne other than acetylene, ethane, if you add water, you end up getting ketone. Remember this. Just by knowing this fact, you can answer it. Say what they're given propane. Then propane means it is not a ethane, it is any other alkyne. Means I should get ketone. Where is the ketone? Observe it. So where is the ketone? Let's take it. Sir, here is the ketone. Any other place? Yes, here is the ketone. Then option 2 and 3 is not possible. Then one more thing. When you are adding water, what you are adding? You are adding H plus and OH minus ion. So first step is formation of a intermediate having this component, H plus or OH minus. But from where you got sulfate? Not at all possible, sir. It has to be answer is 4. Answer is 4. So how this happens, you can write the equation also. I just take a board. Say what you've taken propane, no? CH3, you've taken C triple bond, CH, plus what you're taking water, no? What is water? I told you it is nothing but H plus and OH minus. The first step follows Markovnikov rule. And you know what is Markovnikov rule? The negative part attaches to that carbon having less hydrogen. So this is the intermediate. That's what I told you. You get CH3COH, double bond CH2. And this is a party. That's why I told you the option is fourth. Then what will happen? So then there's something called as tautomerism. So what is tautomerism? Shifting of alpha hydrogen. Shifting of alpha hydrogen, we call it as. Which is alpha hydrogen? This one. This shifts here. What do you get? Then you get CH3, C double bond O, CH3. So what is the option? Yes, sir. Fourth option is the right one. Right. So remember both elaborative way and shortcut also. Once you know the concept, you can go for shortcut. So what is the shortcut? You have to write all the equation. Oh, the shortcut is like this. I will remove this one. I have to clear all these things. The shortcut is what? Just remember, sir, if any ethane is there, and when you add water, you have to get only aldehyde. Sir, any other alkyne plus water, sir, we have to get ketone. That's it. Next one. The element in which electrons are progressively filled in 4f orbitals are called as. You know that one, if electron enters into s orbital, say s block. If they enter into p, we call it as a p block. If they enter into d, we call it as a d block. But if they enter into f orbital, especially 4f series, we call it as lanthanide series. 5f means, so we call it as actinide series. So in this case, it has to be option A. Yes, sir. This is option A. Then again, one more question from the D and F block element. Incorrect statement with respect to cerium is. This I told you in my last class. Look at it. All that questions, previous question also is all from what? D and F block. What is the incorrect statement? I told you, what is a reducing agent? So what is a reducing agent? It reduces others. Reduces others. Means what? So it gives electron. Means what? Undergoes oxidation. So undergoes oxidation. That is called as reducing agent. Means you should give electron. So imagine if cerium is a reducing agent, even if it gives one electron, it will become cerium plus 5. And that is not at all possible, sir. Why? Not possible. Because the oxidation state in a lanthanide is only restricted to +2, +3, and +4. In fact, very common is +3. So cerium +5 is not possible. That means it cannot give electron. If it cannot give electron, it is not a reducing agent. In +4 state, it is not a reducing agent. Not a reducing agent. Yes, in +2 and +3 it can act, but not at +4. So what is the option? He says it is a reducing agent. That is wrong, sir. That's incorrect statement. It can act as an oxidizing agent, but not as a reducing agent. Next one. The mixture of NaCl and potassium dichromate is heated with sulfuric acid. Deep red vapors and formed. Which of the following statement is false? This where you do it in a practical. You do confirmation test for chlorine, no? What you do? You do confirmation test. Test for which acid radical? Chlorine radical. You do it. What do you take? You take, say for example, they have given a sodium chloride salt. You take a salt, you take potassium dichromate crystals, then you add sulfuric acid. And you get red vapors. And the red vapors is due to chromyl chloride, we call it as. What is chromyl chloride? So CrO2Cl2. Cl2. And these vapors are passed through NaOH solution. You get yellow solution then. You get yellow solution. Yellow solution. And this yellow solution, if you add lead acetate, what is lead acetate, sir? CH3COO twice Pb. You get lead chromate. Lead chromate. And lead chromate is a yellow precipitate. Yellow precipitate. So what is wrong? Sir, the first option is wrong. Why? He says you get chlorine. We don't get chlorine, sir. We get chromate. And it gives yellow precipitate. Yes, sir, it gives yellow precipitate. Then it gives yellow solution with NaOH. Yes. The vapors are nothing but chromyl chloride. Yes. But A is wrong, sir, because we don't get chlorine here. This is wrong, sir. Next one. Which of the following statement is wrong? What may be the wrong statement? Let us take one by one. Metals with the highest oxidation states are more stable in oxides than chloride. Yes. Why? The oxygen can have a multiple bonds. So the statement is right. All elements in 3CD, they exhibit variable, yes. But he says, that is wrong, sir. This is wrong. That then this statement may be wrong. Why, sir? For example, zinc. Zinc, they are having a completely filled d orbitals. They cannot show variable oxidation state. So in highest oxidation state, they are acidic in character. Yes, it's right only. Manganese 3 plus, yes, they can act as oxidizing agent. We know that manganese valency can vary from +2 to +7. Anything in between, it can revolve around it. Yes, sir. So what is the option? Option is B, sir. B is the wrong thing. Which among the following is the strongest ligand? Sir, we know that according to spectrochemical series, what is spectrochemical series? We discussed it in a where is, sir, we discussed it in a crystal field splitting theory. And we know that the d orbitals, pi d orbitals are degenerative. They split into two energy level, that is T2G and EG and EG. And the splitting depends on the ability of a ligand. So the splitting depends on the ability of a ligand. So based on that, we have created a spectrochemical series. In that spectrochemical series, CO composite carbonyl groups are very strongest ligand. Next one. So relative lowering of vapor pressure of a dilute solution of glucose is dissolved in 1 kg of water is 0.002. What is the molality of this solution? What is the molality of this solution? What is molality? What is a molality? So molality is nothing but 1 gram molecular weight in 1 kg of solvent. That we call it as a molality. Molality. He says relative lowering of vapor pressure means what? Sir, P naught minus P by P naught, that is called as relative lowering of vapor pressure, is equal to molality. What is the molality? M. So what is M? Sir, M can be written as, molality can be written as what? Number of moles divided by volume in kg. Or we can write it as, so we can write it as P naught minus P by P naught is equal. Number of moles means what? Sir, weight by molecular mass, that is W2 by M2 into, sir, mass, M1 by W1. He has given relative lowering of vapor pressure. How much? 0.002 is equal to, sir, W2 by M2. And he has given what? Weight, 18. And W1, 1 kg of water, he says, that is 1000. Then what is the answer? Give me answer, all of you. Put it, whatever you think, you just write it down. Tell me whether it is A, B, C, or what it is. Sneha, you say B. How much we got it? So I told you 0.002 is equal to W2 by M2. You keep it like that. Into 18 by 1000. No, if you solve it, how much we got? So that ratio we got W2 by M2 means number of moles. How much we got? 0.11 moles, sir. Then you substitute in molarity. What is molarity? Already told you. Molarity is W2 by M2 into 1000 by volume in kg. How much is given? Sir, he has given 1000 only. Volume is how much? 1000 only. Substitute it. What is the number of moles we got? 0.11 into 1000 by 1000. Sir, cancelled. So what is the molarity? Molarity means small m. 0.11 m, sir. That means option A, sir. Option A. Next one. Sir, 1 liter solution of magnesium chloride is electrolyzed completely by passing a current of 1 ampere for 60 minutes in 5 seconds. 60 minutes, 5 seconds. Means what? Sir, we are dealing with chapter electrochemistry. And in electrochemistry, which is the topic? When you say passing the electric current to electrolyte, that means Faraday's law. Is it Faraday's first law or second law? When you say passing through one electrolyte, that means first law, where W we say proportional to Q. Q. What is W? So W is equal to Z into Q, or we can also write it as Z into I into T. We can write it as then what we have to find out? The original concentration of magnesium chloride solution was, what is the concentration? Means we have to give it in terms of M, he says. Capital M. Capital M means we have to calculate in terms of molarity. It's not small m. Small m is molality. This is molarity. Capital M is molarity. That we have to calculate. Then how to do this, sir? This is from Faraday's first law. This we can little bit modify. We need mass first, we need to know. Because if I have to calculate molarity, what is molarity? Molarity is nothing but 1 gram molecular weight in 1 liter. In 1 liter. 1 liter means you can also calculate as M is equal to number of moles divided by, sir, volume in liter. That means what? Again, W2 by M2, weight by molecular mass into 1000. In 1 liter means 1000 by volume. Volume, how much you've taken? Again, 1 liter. He says 1 liter. 1 liter means 1000 ml again. Then for this, first I have to find out this W. How much current is passing? Sir, he is passing current of 1 ampere. How much time? Remember this one. How much time, sir? Time is 16 minutes 5 seconds. Means 16 minutes means you have to convert it into second. That you can take it as 16 minutes into 60 seconds. And what is Z? Sir, this we can modify a little bit. W is equal to Z means electrochemical equivalent. So what is electrochemical equivalent? That means equivalent weight divided by 96,500 into I into T, sir. You substitute. Equivalent weight is 12 divided by 96,500 into I is 1 ampere. Time is, it becomes 965 seconds. If you simplify, it gets cancelled. So 12 into 10 to the power of minus 2 gram we get. This is W, W2. That we have to substitute here. That value we have to substitute here as it. Mass we know. Atomic mass is 24. So it becomes, so 12 into 10 to the power of minus 2. Mass is 24 into 1000 by 1000. That get cancelled. Cancel. So 12 ones are, 12 twos are. 1 by 2 means, sir, it is a 0.5. So 0.5 into 10 to the power of minus 2. And that we can write it as 5 into 10 to the power of minus 3. We can also write it as 5 into 10 to the power of minus 3. Means what? Sir, option is C. Option is C. This is from Faraday's first law. Remember this one. So this is from Faraday's first law. Got it, all of you? If yes, please put it yes. Shirish is not 0.5. Remember, 5 into 10 to the power minus 3. Ujjwal, you're given B. Sneha given B. All of you got it? Good. Sushil, Harsha, got the reply. Thank you. We'll go to next one. A aqueous solution of copper sulfate is subjected to electrolysis using inert electrode. Then the pH of the solution, what will happen to pH of the solution? Let us take copper sulfate. What is copper sulfate? CuSO4. So when you pass electric current, what will happen? It dissociates to form what? Sir, Cu2 plus and SO4 2 minus. And Cu2 plus goes towards cathode. And SO4, they go towards anode. Remember this. In the way you say aqueous, what is aqueous? Water. Water also will dissociate to give what? Sir, H plus ion and OH minus ion and OH minus ion. So as you keep on passing electric current, what will happen? The H plus concentration increases. When H plus concentration increases, that means solution is going to become acidic. When it is acidic, the pH will decrease. pH decreases. pH decreases means what may be the option? Sir, option A. A is the option. Next one. Again from electrochemistry. What is asking? Sir, he is asking E naught. So what may be the option? You have to calculate E naught of Mn plus 7 and Mn plus 4. What he has given? He has given an E naught of Mn plus 7 and Mn plus 2, E naught of Mn plus 4 and Mn plus 2. So let us look at the equation. What is this one? Sir, Mn plus 7, if it has to become Mn plus 2, that means you should accept electron. How many accept electron? You should accept 5 electron. Means it needs 5 moles of electron. 5 moles of electron means it requires 5 Faraday. 5 Faraday. We know that delta G naught is equal to what? So minus NF E naught. NF E naught. Means what? How many electrons are required? Sir, 5 electrons required. That means 5 Faraday. E naught is how much? He has given 1.5. Remember this. Next, we'll go to one more thing. What is the next one? He says Mn plus 4 to Mn plus 2. So Mn plus 4 should become Mn plus 2, no? Plus 2. That means he should accept 2 electron. Means 2 moles of electron. It requires 2 Faraday. So then the equation becomes delta G naught is equal to minus NF E naught. That means, sir, minus 2 Faraday. E naught how much is given? Is given 1.2. 1.2. And what is asking? Sir, he is asking when Mn plus 7 becomes what? So Mn plus 7 becomes Mn plus 2. Sorry, Mn plus 4. Mn plus 4 means he has to accept 3 electron, 3 moles of electron. That means 3 Faraday. 3 Faraday. In this case, delta G naught is equal to minus 3 of E naught. Because we don't know the E naught. He has not given the E naught value. That's what we have to find out. That's what we have to find out. Then what we can do, we can combine this equation 1 and equation 2, and then we can compare with equation 3. Equation 3. So it becomes how much? So equation 3 is what? I already told you, minus 3 of E naught is equal to equation 1. How much it is? How much it is? Then you get the answer over. How much it becomes? Look at what I have written. 5F 1.5. 5F 1.5 means how much it is? So minus 7.5 Farad. Plus, here in second equation I have written 2F 1.2. 2 into 1.2 means how much? So it's a 2.4 Faraday. Now easily you can calculate E naught. So what is E naught then? Sir, E naught is equal to around, it comes to around 1.7 volts. 1.7 volts means, sir, option A. Just remember this one. Equation 1, 2 should be compared with equation 3. This is equation 3, sir. This is equation 1, sir, plus equation 2. What will happen? So the Faraday gets cancelled. And what we left with by E naught? E naught is equal to 1.7 volts. Next one. The plot of T and of versus R naught for a reaction is a straight line parallel to x-axis. The unit for the rate constant of this reaction is, means what? He says a plot of T and of versus R naught. R naught means initial concentration will give you straight line. Means what? That means T and of does not depend on initial concentration. Depend on initial concentration. And then, sir, this may be first order reaction. What is first order reaction? So K is equal to 2.303 by T log of R naught by R. So the concentration term gets cancelled. So then means 1 by T. 1 by T means what? So it may be per second. Per second means the option B, it has to be. Should be. What should be the option? So it has to be the B. Let's check it out one by one. We'll check. You know that for zero order reaction, what is the K? K is equal to R naught by R naught minus R by K, T. And T end of life depends on the initial concentration when zero order. Whereas in first order, the half life does not depend on the initial concentration. That means it is a first order reaction. It's a first order reaction. Next one. The mass of AgCl precipitated when a solution containing 11.070 gram of NaCl is added to a solution containing 3.4 gram of AgNO3. So what is the equation that we'll write? First, he says AgCl precipitated Yavaga when NaCl is added to that. NaCl is added to AgNO3. That we'll write. So what you get? Sir, you get AgCl plus NaNO3. So this is our equation. AgCl is a white precipitate. So how the equation? Sir, 1 mole of NaCl combines with 1 mole of AgNO3 to give 1 mole of AgCl and 1 mole of NaNO3. Remember this. So what is 1 mole? It's a molecular weight. 1 mole means what? It's molecular weight. What is the molecular weight of AgNO3? Sir, what is Ag? Sir, he has given. Ag is 108. 108. So nitrogen, sir, 14. So 3 oxygen atom. 3 oxygen atom. So how many times you have to write? 16, 16, 16, 3 times. So how much it comes? It comes out to be 170. That means 1 mole means 170 gram of AgCNO3. AgNO3. How much amount of AgCl it gives? Again, AgCl. Ag is how much? 108, sir. Chlorine is how much? 35.5. That means what is its molecular weight, sir? 143.5. That means it gives 143.5 grams. So if you take 170 gram, it gives you 143.5 grams of HCl. And in the given equation, how much grams you have taken? 3.5 grams. How much it gives? We don't know, sir. So X is equal to what? Cross multiplication. 3.4 into 143.5 divided by, sir, 170. 170. That's what you have to do. Find out what is the answer. Harsha, NCRT book is enough. NCRT is enough for our CET and NEET. Don't worry about any other book. Don't worry about another book. If you solve all the exercise in NCRT, please solve the exercise. And also, please go to exemplar book. If you don't have exemplar book, it is freely available in a net as a form of PDF. Download the PDF and solve it. That's more than enough. If you have a NCRT solution, it may be around 100 rupees. That's it. NCRT solution. Sometimes in a net, it is available freely. Take NCRT solution book. Solve all the problems in exemplar book. Don't forget. Don't forget. Now what is the answer for this one? How much it comes? Sir, it comes out to be around 2.87 gram or 2.8. 2.8 means, sir, option. Yes, sir, option. Next one. Most of the questions, no. If you look at all previous year question paper, I'll tell you, there is no question out of NCRT. Till to date, no question out of NCRT. Everything is based on NCRT. But many of us, we don't solve the exercise only. We don't solve. There is in-text questions. We neglect it. Solve that in-text questions. And then NCRT exemplar, don't leave it. These things first you finish it before you study any other book. Sir, is it enough? I'll tell you more than enough. I'll tell you more than enough. Sir, whether I have to follow some other books, so whether I have to study extra, not a penny more, not a penny less. I will tell you, very simple, not a penny more, not a penny less. Means what? This, our NCERT book is enough. Is enough. Enough. And don't forget NCERT exemplar. Sometimes many questions come directly from that. Next one. Say two particles A and B are in motion. This is from structure of atom. And wavelength associated with A is 33.3 nanometer, and the wavelength associated with B is B, whose momentum is 1/3, he says. Momentum, the P, is nothing but what, sir? Is a lambda A wavelength by lambda B is equal to PB by PA. And then what it becomes? 33.33 divided by lambda B, we don't know. And he says 1/3. Then this is 1. Then what is the option? Sir, option is, which can be the answer there? This almost comes to what? So 33 point by 3, that is almost 1. 1 into 10 to the power of minus 7. That means our option is B, sir. That 0.99 you can take it as just 1. So the first ionization enthalpy of the following element are in the order. What is ionization enthalpy? Energy required to remove the electron. I told you left to right, sir, it increases. So which of the elements you are having, sir? You are having carbon, that is group 14. So nitrogen is there, that is group 15. So among carbon and nitrogen, definitely nitrogen is more. So everything should be less than nitrogen. Then you are having silicon, where it is present, that is below silicon, carbon. Phosphorus, that below phosphorus, nitrogen. So when you take the carbon and phosphorus, the carbon is less than nitrogen and phosphorus is less than carbon. Among silicon and phosphorus, silicon is less than phosphorus. So what is the order? This, sir, option B. Solubility of AgCl is least in, where it is? Is least in. So what is AgCl? So AgCl in the solution dissociate as what? Ag plus and Cl minus. And if you take any of this salt, for example, I will take aluminum chloride. Why you have taken aluminum chloride? Because it gives more Cl minus ion. It gives you Al3 plus and it gives you how many moles of Cl minus? 3 moles of Cl minus. So what is happening? Sir, here the product concentration is increasing. The product concentration increases. When product concentration increases, what will happen? The equilibriums are shift towards left side. Left means that it becomes a backward reaction. So solubility is a forward reaction. Backward means the solubility of AgCl decreases. Due to what? We can also call it as common ion. Due to, sir, common ion effect, the dissociation of AgCl decreases. When the dissociation decreases, solubility decreases. This happens because of AlCl3. Where is present? Sir, B is B. Which of the following equation does not represent the Charles law? Does not represent Charles law. What is Charles law? We know that according to Charles law, volume is proportional to temperature. Or we can also write V by T is equal to circle. Or we can write V is equal to K into T, or V by T is equal to K, we can write. Or if we take, we can write it as, sir, log K is equal to log V by T. This can also be written as log K is equal to, in logarithmic terms, log A by B can be written as log A minus B. Like that, log V minus log T we can write. So what option is wrong here? Sir, when you say log K is equal to, so it is a log B minus log T. But as in option A, sir, he has given plus. So A is wrong. So then that is our option. That is our option. Which is the most suitable reagent for the following conversion? Most suitable for the following conversion. So what is happening here? If you have to observe, if you observe very carefully, here is a ketone. Ketone has become a carboxylic acid. But if you observe carefully, how many carbon you add? 1, 2, 3, 4, 5, 6 you add. But here how many carbon is there? Only 5 carbon is there. That means 1 carbon is removed. 1 carbon is removed. So 1 carbon is removed. Which of the reagent can do that? Say for example, Stalin's reagent can react only with aldehydes. So it cannot be possible. Tin and NaOH is a reducing agent, means they add hydrogen. They add hydrogen, sir. They are not oxidizing agents. They cannot be possible. So left with only 2 options, benzyl peroxide and iodine and NaOH. Peroxide gives you free radical and they are not very strong. So they are not be possible. So only is iodine and NaOH. What is this? It is nothing but iodoform reaction. What is iodoform reaction? When it reacts with any compound having general formula RCOCH3, only this compound can undergo iodoform reaction. What is that? This one, CO group, carbonyl group next to methyl. Yes, sir. Here you are having COCH3, then it gives iodoform reaction. What will happen when it reacts with iodine and NaOH? This part, the last part is there, no CH3, that combines with iodine and becomes CHI3. So here 1 carbon, I told you, 1 carbon is removed. That as carbon has become iodoform. Iodoform is a yellow color, yellow precipitate. So your compound from 6 carbon, it becomes what? 5 carbon. 5 carbon. Because the bond will break here. Break here. Next one. Which of the following is the least soluble in water at 298 Kelvin? Least soluble in water at 298 Kelvin. Solubility, remember, always directly proportional to how many bonds it forms. How many hydrogen bond with water? With water. So these are all primary amine. They can form how many hydrogen bonds? So they can form 3 hydrogen bonds. Yes, 3 hydrogen bonds. This can form 2 hydrogen bonds. But here, sir, there is no hydrogen only. This is least soluble. This is least soluble, sir. That means it should be option D, sir. Option D. Next, if aniline is treated with, means if it undergoes nitration, it gives para and meta. Why? Because if aniline undergoes nitration, it is supposed to give only ortho and para. So if this is the aniline, what is aniline? Benzene with NH2 group. If it undergoes nitration, what is nitration? Addition of NO2 group. It is supposed to give only ortho and para. Why, sir? Because NH2 is a activating group. And activating group will give only ortho and para. What is ortho? Either position 2, para means position 4. But he says, here, you're also getting meta. Why it happens? Why it happens? Then you have to tell this happens because some of the few aniline molecule means NH2 undergoes protonation with nitric acid. What is nitric acid? Is acid. It gives H plus. Is acid. It gives proton and undergoes protonation to form anilinium ion. Sir, NH3 plus. We call it as anilinium ion. And NH3 plus is a deactivating group, sir. And deactivating group always sends incoming electrophile to meta position. Incoming electrophile to meta position, sir. So just look for these words. Protonation, sir, anilinium ion, where it is present. If you observe it, yes, sir, we can see it. Where we can see it? I told word, sir, in the last option. Protonation of NH2, which causes deactivation of benzene ring due to presence of NH3 plus. So option is D, sir. Option is. Yes, sir. Option is D. In nucleic acid, nucleotides are joined together by, we know that it's a phosphodiester linkage. Option D. Which are following is a generally water soluble? Insoluble, sorry, water insoluble. Amylose, we know that it is a component of starch. It is soluble because it's linear. Glycines are amino acid it is. And amino acids are soluble in water. Yes, are soluble. Vitamin C is a water soluble vitamin, sir. So only option is so 5%. Which of the following possess net dipole moment? Means it has some kind of a dipole moment. What is dipole moment? Sir, we say mu is equal to charge into distance. We can measure the polarity. So in case of barium chloride, no, there is no dipole moment because in this case, dipole moment of this bond gets cancelled by the opposite bond. So the net dipole moment is equal to 0. So we should not get mu is not equal to 0. CO2, so that is also linear. Here also mu is equal to 0. SO2, let us understand. Yes, sir, this is a bent molecule. Bent molecule. So what will happen? So mu is not equal to 0. The dipole moment is not cancelled. BF3, sir, here also the bond will cancel each other. The polarity gets cancelled each other and mu is equal to 0. Then option should be SO2. Let's check it out. Yes, sir, answer is SO2. The number of sigma bond and pi bonds in naphthalene is how many sigma bonds and pi bond in naphthalene? What is naphthalene? Naphthalene is nothing but benzene ring. There will be a 2 benzene ring. That's it. So totally how many pi bond? So 5 double bond is there. So that may be 5 pi bond. 5 pi bond. But how many sigma bonds? Sir, here, if you observe it, all these joints are what? Carbon. and you'll be having hydrogen here. Everywhere you'll be having hydrogen. It's understood. Here also hydrogen. And now you calculate how much it is. Sir, 1 sigma bond, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, and 19, sir. So then 19 sigma bond is there. That means 5 and 19 means option D, sir. Option D is the answer. The reaction in which delta H is more than delta U is where it can be. What is delta H? Sir, enthalpy. Enthalpy is equal to what? Sir, internal energy, that is delta U plus, sir, delta NG/RT. So what is delta NG? Delta NG is nothing but gaseous moles, gaseous product minus gaseous reactants. Reactant. Only when delta Ng is equal to positive, then in that case delta H becomes more than delta H. Where it is possible. Just check it out, all the equation. Sir, here let us take. In first equation, the product is 2 moles minus how many reactant is there, sir? There are 3 moles of hydrogen, 1 mole of hydro, 1 mole of nitrogen. So it is 2 minus 4, sir. That is, it becomes minus 2. So this cannot be the answer. Let us go to C. How many moles here? Product is 2 minus, and reactant is 1 plus 1, sir. Again 2 minus 2. It becomes 0, sir. It cannot be. Let us go to B. So what are the products here? Liquid we cannot take. Only we have to take gas. So 2 minus, how many moles of reactant? 2 moles of oxygen and 1 mole of methane. There is 2 minus 3. That becomes minus 1. So this is also not possible, sir. So what is remaining? That should be the answer. Let us check it out. Solid, we cannot take solid, sir. We have to take only gas. How much? 1 mole. And this is also solid. That is 1 minus Z. 0 is equal to 1, sir. So only in case of D, sir, delta H is more than delta U, sir. The number of moles of electron required to reduce dichromate to Cr plus 3 is how much it is? What is dichromate? Sir, Cr2O7 2 minus is converted into what? Cr plus 3. How many Cr plus 3? 2 Cr plus 3. And here, what is the oxidation number of oxygen, the chromium? So it is a plus 3. And here, how much it is? Of chromium. So we say plus 6. So how come you got to know? We know that in a redox reaction we have calculated. How to calculate? Say Cr2O7 minus 2 the H1. Now this is ion. So the total oxidation number can be taken as minus 2. So imagine we don't know chromium. Then let's take it as chromium as X. 2 chromium is 2X. And oxygen is minus 2, we know that. And 7 2s are 14. That is 2X is equal to 14 minus 2. That is equal to 12. That means X is equal to plus 6. That's what I have written here. If plus 6 has to become plus 3, if plus 6 has to become plus 3, how many electrons should accept? So we should accept 3 electrons. So 1 mole of dichromate ion needs how many electrons? 3 electrons from 1 chromium. So there are 2 here, no? So it needs 6 electrons. So he has given how much? 0.2 mole. So 0.2 mole needs how many electrons? We don't know. X. So X is equal to 0.2 into 6. 6 2s are 12. There is 1.2, so 1.2 means option C. So got it, all of you? All of you put yes if you have understood it. Many times this kind of question is there. These questions will combine 2 chapters, basic concepts of chemistry as well as redox reaction. All of you, please put it as yes. If you're not able to understand, put no also, no problem. But I want answer. All of you should give answer. Till then, I will not go ahead. Yeah, very good. Sushil, thank you. Anonymous, thank you. Very good. Abhishek, once again, sir. No problem, I will just tell you. Charmila Reddy, thank you. Look at here, Abhishek. This is for you. I will take another color. I'll get black color. So what you have to do? First, you have to find out the oxidation number of chromium here. How to find out? This is how I have done. The total oxidation number of a dichromate ion is -2. N charge is adine barkobek. That we will write it as -2. So there are 2 chromium atoms, so I have taken just 2x. So how much I got? +6. That's what I have written here, +6. And he says dichromate is converted into Cr+3. The charge on the ion itself is the oxidation number. So if +3, 6 has to become +3, how many electrons you require? 3 electrons. But there are 2 Cr. So you take it as 2 into 3. Totally, how many electron is required? 6 electron is required. For 1 mole of dichromate, we need 6 electron. So he has given for 0.2 moles. So I have done a cross multiplication. That's it, nothing else. Got it, Abhishek? Abhishek, got it? Anonymous, got it? Yeah, okay then. No problem. Next one, we'll go for next one. I already have given answer. Boric acid. What is boric acid? The BOH thrice. Functions as is killing. BOH means what? BOH thrice. This is BOH thrice. So totally, how many electrons is having around that? 6 electrons. And to reach octet, And to reach octet, he needs electron. Means he has to accept electron. Anybody who accepts electron is called as what? Lewis acid. Then option is B, sir. Match the following. Match the following pK values. So how to do this one? Very simple. We know that if pK is less, that means more acidic. And if pK is more, opposite this is less acidic. First, we have to find out who is less acidic, who is more acidic. Then it becomes easy. And always remember, phenol is stronger than alcohol. And alcohol are weaker. It's a weak acid, alcohol. Which is alcohol? Ethanol is a weak acid. That means you should have a highest pK value. Less acidic means, remember, pK should be more. So ethanol should be 16. C should match with 1. C should match with 1 means there's only one option, option C. Without doing others, you can got the answer. Let us check it out. Is it C? Let me check it out. Yes, sir. C is the answer. Then who is more acidic, sir, among all these things? Picric acid is more, even is less acidic, no? Less acidic means more pKa. Who is more acidic? Picric acid is more acidic. Why, sir? What is picric acid? A phenol with more NO2 group. And NO2 is a withdrawing group. Withdrawing group increases acidic nature. Sir, increases acidic nature. So it's more acidic. More acidic means less pKa. That means you should match with second. D should match with second. Yes, sir. It is proper only. So C is the answer. Which of the following can be used to test the acidic nature of ethanol? What we can take? Ethanol is acidic. Remember this one. And show it as acidic, we have to treat with sodium metal. Say phenol. How do you show phenol is acidic? Very simple. If it reacts with sodium metal, it is acidic. How do you show that carboxylic acid is acidic? If it reacts with sodium metal, it is acidic. And if they react with NaOH, then they are acid. Ethanol will not react, whereas phenols and RCOH will react. So they are acid. They are acidic as well as acid. Only acid will react. And NaCO3 will react. Only carboxylic acid will not react. Phenol and ethanol will not react. Because only strong acids will react. What is the option again? Sir, option is to show it is acidic, we need only sodium metal. Option B, sir. Next one. What may be the option? A, B, C. If you observe the molecule, so what is happening? Here, if you observe here, in the option A, CHO has become CH2OH. Means, so it is a reduction. It's a reduction. Oxidation means it could have become carboxylic acid, reduction. And which is the reducing agent? Sir, either you have to take sodium borohydride, either you have to take palladium. Only these two people are the reducing agent. But double bond remains as a double bond. So I cannot use double palladium. I should use NaBH4. So A is NaBH4. Sir. Then come to B. What is happening in B? If you observe in B, the CH2OH has become aldehyde. Again, this is oxidation. So this is oxidation. But it is not complete oxidation. So it's a controlled oxidation. And we can do controlled oxidation. Sir, only remember, we can do controlled oxidation. Only PCC can do it. So B should be PCC, pyridinium chlorochromate. Then we'll go to one more thing. So C, what is happening in C? If you observe it, the aldehyde has become alcohol. Again, reduction. And also, if you observe it, the double bond has become single bond. Again, reduction. And this kind of reduction is done only by palladium. So C should be palladium. So what is the option then? So then option is yes, sir. Option is yes. Next one. Propanoic acid undergoes a user reaction to give chloropropanoic acid. The product obtained is. What may be the option? What is propanoic acid? So 3 carbon atom, CH3, CH2, COH. And what is chloropropanoic acid? Cl, CH2, CH2, COH. This is propanoic acid. And remember, always the presence of electron withdrawing group we call it as EWG. Sir, increases acidic nature. Acidic nature. So the option C is right, sir. It is stronger than propanoic acid. Stronger than propanoic acid. Next one. Sir, R and S forms a benzyl benzoate when treated with each other. Ian speaks. First, he says R and S forms a benzyl benzoate. Means if they react with each other, you get a benzyl benzoate. What is benzyl benzoate? Means you're having a benzene. What is benzoate? That means you're having a ester linkage. COO. Again, you're having benzene. This is we call it as a benzyl benzoate. Then this is ester. If this has to be ester, then your R should be benzoic acid. R should be benzoic acid, sir. Then S should be alcohol. It should be alcohol. What alcohol? CH2OH. And remember, everything benzene is common. So you have to get carboxylic acid and alcohol means it has to be undergo Cannizzarose reaction. It has to undergo Cannizzarose reaction. And who can undergo Cannizzarose reaction? Only aldehydes without alpha hydrogen, like benzaldehyde can undergo Cannizzarose reaction. And how to get benzaldehyde? By Rosenman reduction. By Rosenman reduction. What is Rosenman reduction? Then you have to take benzyl chloride. So what is benzyl chloride? One with the COCl group This we can also write it as T6H5 COCl. That means option A, third option is the answer. Because what is a kinase reaction? Only aldehyde having no alpha hydrogen atom. They undergo kinase reaction with concentrated NaOH. NaOH to form what? A carboxylic acid and a alcohol. Yeah. Next one. Very good. Most of you given A, right? Next one. Which of the following is a network crystalline solid? And you know that this comes from chapter solids. And solids have been classified into 3 criteria. Based on what? Sir, magnetic property. As what? Sir, diamagnetic. Then ferromagnetic. Then paramagnetic, like this. Sir, based on conductivity, they have been classified as what? You know that one. Conductor, insulator, semiconductor. And semiconductor is of N type to P type. And there's one more classification based on types of particle in a lattice point. Types of particle. And based on this, you are having 4 types of solids. Sir, ionic solid, if ions are the present in lattice point. Molecular solid, if molecule is present, sir. Sir, if atom is present, covalent. So if metal is present, metallic. So NaCl is ionic, sir. So it cannot be the answer. Ice is molecule, polar. In there you are having polar and non-polar. So this cannot be answer. So which is the network crystalline solid? Sir, aluminum nitride. That is also called as covalent. Then again, one more problem on solids. The number of atoms in a 2.4 gram of a body-centered lattice with a edge length of 200 picometer. Hagadre, how many atoms are present? How to do it, sir? How to do it? We know that mole concept. Look at it. He has combined our solid and basic concepts of chemistry. What is that? How he has combined? You know that, sir? 1 mole. 1 mole means its molecular weight will contain Avogadro number of particle. If we know 1 mole, then we can calculate. Means its molecular weight, if you know, we can calculate easily. Why, sir? Just cross multiplication. Then 2.4 gram contains how much? But first we should know M. But so how to find out M? We know that density is equal to what? Density is equal to, sir, number of particle in a unit into molecular mass. Here is our M. Divided by A cube into Na. So what we need to find out, sir? We need to find out M. So then M is equal to what? M is equal to, so density into A cube into Na divided by, so what it becomes, that, sir? If you do this, how much you get? Sir, I get 24. So this is the molecular weight. That put it here. Oh, 24. So cross multiplication. What is cross multiplication? X is equal to 2.4 into 6.02 into 10 to the power of 23 divided by, so 24. How much it becomes? So 2.4 gets cancelled. It becomes 6 into 10 to the power of 22. What is the option? Then option is C, sir. Option is C. Next one. So 1 mole of sodium chloride is doped with 10 to the power of -5 moles of strontium chloride. The number of cationic vacancy in the crystal lattice is. So what is telling is that in a sodium chloride crystal lattice, what you'll be having then? You'll be having Na plus and you'll be having Cl. This is the lattice system, what you have. But when you dope with strontium, what is strontium? Strontium will have 2 positive charge. So one strontium can satisfy 2 Na plus ion. 2 Na plus ion. That means strontium can take the place of a 2 Na plus ion. So what is happening? Because of this, there is a 1 vacancy now. So there is now 1 vacancy where here you can see this is 1 vacancy. This is called as cationic vacancy. So 1 strontium ion will create 1 cationic vacancy. So how many you are taking? Say you are taking 10 to the power of -5 mole. It will create that much cationic vacancy. Cationic vacancy. So then how much it becomes? In a 1 mole, we are having how much? 6.02 into 10 to the power of 23, no? Into 10 to the power of -5. So how much it is? 6.02, sir, into 10 to the power of 18, it becomes, 18 becomes.
Sir, it's time up.
Yeah, we can conclude here, no problem. Dear students, thank you all. As I told you, now not to worry too much about how much you have prepared and how much you have studied and all those things. What you can do is that now already today is 10th. Okay. Whatever you have studied, now don't go for new portions, or if you're not studied, not to worry. As I told you, here what is important is don't leave any second PU chapter because we have 45 questions. And in the first PUC, I told you there will be around 15 questions. It may range, it may vary. And in first PUC also, I told you what are the important chapters. Don't leave chemical bonding. And then don't leave chapter 12. What is the chapter 12? GOC, organic chemistry, don't leave it. Then chapter 13, that is hydrocarbons, don't leave it. These 3 chapters will give you maximum questions. In this 15, I think sometimes you may get around almost 6 to 9 questions in a year only from these 3 chapters. Then, Bagli sir, 1 minute, shall I take?
Yes, sir. Yes, sir.
And then, please don't leave one more chapter. What is that, sir? That is equilibrium. Yeah. And if time permits, thermodynamics also. So these chapter, and I told you classification. All chapters you should study, but on priority, first complete all these chapters in first PUC. Second PUC, don't leave any chapters. Don't leave any chapters. And what you do, I told you, what I've done. Just practice chapter wise. Chapter wise. And last, you take up some mock exams. And our university will be conducting mock exam. I think Bagli sir will give you all the information. Okay. Don't leave this mock exam. That is what giving you edge over others. Now, whatever you have studied, that's over. That's why I'm telling. Now, don't worry about reading textbook and everything. Don't do it. Now is not a time to read textbooks. Please don't do that. Use your notes, whatever the notes you made, whatever the concept map you have made, whatever the revision notes you made, make use of it. And now, immediately get into mock exam mode. Practice mock exam as much as possible. I think our university will be doing a lot of mock exams. Don't leave it. It gives you a very good edge over others. Edge over others. And our university will give you informations. They do it online, so it becomes easy for you. Practice a lot. Practice a lot. And if anything is there, don't think that, oh, we are not taking class now, we stop it. So means chemistry. Tomorrow you'll be having biology and physics. Bagli sir will give you information.
Yes, sir.
Okay. Even if you have some questions on chemistry, something like that, okay, please inform Bagli sir. I can answer it also. I can answer it. Okay, do well. Okay, all the best for every student. Thank you all. Bagli sir, thank you very much.
Thank you. Thank you so much, sir.
Yep.
Dear students, tomorrow also you will be having class 4 to 8. Don't miss it. These end classes are very good revision. Please do not miss. Do join. I will give you the information in the morning. And recordings I'll be sending regularly. Dear students, all the recordings will be available in the same link. You need to just scroll back and see. All the recordings will be available in the same link.